In September 2024, the Australian Parliament passed legislation that sent shockwaves through the scientific community worldwide. The Genetic Heritage Act 2024, scheduled to take full effect in January 2025, created the world's first legal framework for de-extinction, the deliberate resurrection of extinct species through genetic engineering. But this wasn't the Jurassic Park scenario that had dominated popular imagination for decades. The law contained strict limitations that immediately ruled out dinosaurs, mammoths, and other ancient extinctions. Instead, it focused exclusively on species that had vanished within the last 50 years, creatures whose completeness sequences had been preserved, whose ecological niches still existed, and whose revival could potentially restore damaged ecosystems. The first two candidates were already identified: the Thylacine, Tasmanian tiger, extinct since 1980, and the gastric brooding frog, extinct since 1960. Both had well-preserved genetic material. Both had close living relatives that could serve as surrogates. Both had habitats that remained largely intact. And both represented not just scientific challenges, but profound ethical questions about humanity's relationship with nature. The law didn't just permit de-extinction. It regulated it, establishing oversight committees, requiring ecological impact assessments, mandating indigenous consultation, and prohibiting commercial exploitation. This wasn't a free-for-all. It was a carefully crafted framework designed to balance scientific ambition with ecological responsibility and ethical restraint. And it was about to transform conservation biology forever. The Genetic Heritage Act emerged from decades of scientific progress and ethical debate. The concept of de-extinction had existed since the 1980s, when advances in cloning technology first made the idea theoretically possible. The successful cloning of Dolly the sheep in 1980 demonstrated that adult somatic cells could be reprogrammed to create new life, a breakthrough that immediately raised the question: could we do this with extinct species? The answer, initially, was no. Cloning requires intact living cells, and extinction by definition means no living cells remain. But the field of ancient DNA research was advancing rapidly. Scientists learned to extract and sequence DNA from museum specimens, frozen remains, and even fossils. By the early 2000s, complete genomes had been sequenced for species like the woolly mammoth and the Neanderthal. The problem was that ancient DNA is always fragmented and damaged, like a book with most pages torn out and the remaining pages covered in coffee stains. Reconstructing a complete, functional genome from these fragments seemed impossible. Then came CRISPR. The gene dating technology, developed in 2001, revolutionized genetic engineering by allowing scientists to precisely cut and paste in the sequences with unprecedented accuracy and efficiency. Suddenly, de-extinction didn't require perfect ancient DNA. It required a close living relative whose genome could be edited to match the extinct species. The template was already there. Scientists just needed to rewrite specific sections. This approach, called genetic rescue, rather than true resurrection, made de-extinction feasible for the first time. But feasibility didn't equal wisdom. Scientists, ethicists, conservationists, and indigenous communities debated whether we should revive extinct species, even if we could. The Australian law represented the first attempt to answer the question through legislation, rather than leaving it to individual researchers or private companies. The specific provisions of the Genetic Heritage Act were carefully calibrated to address these concerns. Section 1 established the 50-year rule: only species that went extinct after 1980 could be considered for revival. This wasn't arbitrary. DNA degrades over time, with a half-life of approximately 20 years under ideal preservation conditions. After 50 years, even well-preserved specimens contain significant DNA damage, but not so much that CRISPR editing becomes impossible. More importantly, ecosystems change over time. A species that vanished 50 years ago likely left behind an ecological niche that hasn't been completely filled by other organisms. A species that vanished 500 years ago almost certainly did not. The law recognized that de-extinction without a place to belong would be cruel and ecologically meaningless. Section 1 required that any revived species must have a viable ecological pathway, a scientifically validated plan for reintroduction into the wild, including habitat restoration, threat mitigation, and population management. This provision prevented de-extinction from becoming a zoo curiosity or private collection item. Section 1 mandated consultation with indigenous communities whose traditional lands or cultural heritage would be affected by revival efforts. For the Thylacine, this meant engaging with Aboriginal Tasmanian groups who had coexisted with the species for thousands of years before European colonization. Section 1 Euclid prohibited commercial exploitation of revived species or their genetic material, ensuring that de-extinction would serve conservation and scientific purposes rather than profit motives. Section 1 Euclid established an independent oversight committee composed of geneticists, ecologists, ethicists, indigenous representatives, and animal welfare experts to review and approve each de-extinction proposal on a case-by-case basis. This committee would evaluate not just scientific feasibility, but ecological impact, ethical considerations, and public benefit. The law was comprehensive, cautious, and unprecedented. But why Australia? Why not the United States, China, or the European Union, countries with larger scientific budgets and more advanced biotechnology sectors? The answer lay in Australia's unique ecological crisis. The continent has the worst mammal extinction rate in the world, with over 1/4 native species lost since European settlement in 1916. Many of these extinctions occurred recently and were directly caused by human activities, habitat destruction, invasive species, hunting. The Thylacine was perhaps the most iconic example. Once widespread across mainland Australia and Tasmania, the striped marsupial carnivore was systematically eradicated by European settlers who viewed it as a threat to livestock. The last known Thylacine, named Benjamin, died in captivity at Hobart Zoo on September 1988. His death marked not just the loss of a species, but the failure of conservation efforts that had come too late. For decades, the Thylacine became a symbol of human-caused extinction, a ghost haunting Australia's ecological conscience. Reviving the Thylacine wasn't just a scientific challenge. It was an act of atonement. It was an opportunity to correct a historical wrong, to restore a missing piece of Australia's ecological heritage, and to demonstrate that humanity could learn from its mistakes. The Genetic Heritage Act was, in part, a response to this moral imperative. It acknowledged that humans had caused these extinctions, and therefore had a responsibility to explore every possible avenue for restoration. But it also recognized that responsibility required restraint. Revival wasn't an automatic right. It was a privilege that had to be earned through rigorous scientific review, ecological planning, and ethical consideration. The international reach into Australia's law was swift and defiant. Conservation organizations like the World Wildlife Fund and the International Union for Conservation of Nature, IUCN, issued cautiously supportive statements, emphasizing that de-extinction should complement, not replace, traditional conservation efforts to protect living endangered species. They warned against conservation moral hazard, the risk that the promise of future revival would reduce urgency to prevent extinctions happening today. If people believe we can simply bring species back later, they might become complacent about habitat destruction, climate change, and other threats. This was a legitimate concern. The law addressed it by requiring that de-extinction projects demonstrate net positive impact on overall biodiversity conservation, not just the revival of a single charismatic species. Biotechnology companies, particularly those in the United States and China, expressed interest in the Australian framework but also frustration with its restrictions. Some argued that the 50-year rule was arbitrary, and that advances in synthetic biology would soon make older extinctions feasible. Others questioned whether government oversight would stifle innovation and drive research underground or to countries with laxer regulations. Indigenous groups worldwide watched closely, recognizing that Australia's approach to consultation and cultural heritage could set a precedent for similar laws in their own countries. Some expressed support for the emphasis on indigenous knowledge and consent. Others worried that even well-intentioned revival efforts could disrupt cultural relationships with extinct species that were meant to remain in the spiritual realm. The scientific community was similarly divided. Geneticists and molecular biologists generally welcomed the law as a necessary step toward responsible innovation. Ecologists and field conservationists were more skeptical, questioning whether revived species could truly re-establish wood populations or would remain dependent on human management indefinitely. Ethicists debated whether de-extinction represented hubris or humility, whether it was an arrogant attempt to play God or a humble effort to repair damage we had caused. These debates were healthy and necessary. They reflected the complexity of the issue and the high stakes involved. The Australian law didn't resolve these debates. It created a framework for having them in a structured, transparent way. The timing of the law was also significant. 2024 marked the halfway point of the United Nations Decade on Ecosystem Restoration. 2030, a global initiative to prevent, halt, and reverse ecosystem degradation worldwide. De-extinction, when done responsibly, could contribute to this goal by restoring lost ecological functions. The Thylacine, for example, was Tasmania's apex predator. Its absence had cascading effects on the ecosystem: predator release, increased numbers of smaller predators like feral cats and foxes, altered prey populations, and changes in vegetation structure. Reintroducing a Thylacine-like predator could help restore balance to Tasmanian ecosystems, benefiting countless other species. Similarly, the passenger pigeon, once the most abundant bird in North America with flocks numbering in the billions, played a crucial role in forest ecology through seed dispersal and disturbance. Its revival could help restore eastern North American forests that had changed dramatically since its extinction in 2000. These ecological arguments gave de-extinction a conservation rationale beyond scientific curiosity or technological demonstration. But they also raised difficult questions. How do we know what ecological role a revived species will actually play? Ecosystems have changed in the decades since these species vanished. Climate change has altered habitats. Invasive species have established themselves. Human land use has fragmented landscapes. A revived Thylacine wouldn't be returning to the Tasmania of 2000. It would be entering a different world, one shaped by human activity in ways that might not accommodate its needs. The law recognized this uncertainty and required adaptive management plans that could be adjusted based on monitoring and research. De-extinction wasn't a one-time event, it was a long-term commitment to learning, adapting, and stewarding revived species and their ecosystems. The Genetic Heritage Act also reflected a broader shift in conservation philosophy, from preservation to restoration, from protection to active intervention. Traditional conservation had focused on protecting existing species and habitats from further harm. This approach had saved countless species from extinction, but it had also failed to prevent many losses. The current rate of extinction is estimated to be just about 9 times higher than the natural background rate, driven primarily by human activities. Some conservationists argue that passive protection was no longer sufficient. Active intervention, including genetic rescue, assisted migration, and even de-extinction, was necessary to address the scale of the biodiversity crisis. This interventionist approach was controversial. Critics warned of unintended consequences, ecological disruption, and the hubris of assuming we could predict and control complex natural systems. Proponents countered that we were already intervening in nature through habitat destruction, climate change, and species introductions, often with devastating consequences. The question wasn't whether to intervene, but how to intervene wisely. De-extinction, when done responsibly, could be part of that wise intervention, a tool in the conservation toolkit rather than a silver bullet. The Australian law embodied this nuanced perspective. It didn't mandate de-extinction. It permitted it under strict conditions. It recognized both the potential benefits and the significant risks. It created a framework for careful, evidence-based decision-making rather than ideological positions. This balanced approach was what made the law groundbreaking. It wasn't perfect. No legislation is. But it represented a serious attempt to grapple with one of the most profound questions facing humanity in the Anthropocene. Having caused so much extinction, do we have a responsibility and the capability to bring some species back? The law's passage also reflected changing public attitudes toward extinction and conservation. For much of the 20th century, extinction was viewed as a tragic but inevitable consequence of progress. Species were lost to development, agriculture, and resource extraction, and while conservationists fought to save them, the broader public often saw these efforts as sentimental or obstructive to economic growth. But in recent decades, public awareness of the biodiversity crisis has grown dramatically. Documentaries like Our Planet and David Attenborough, A Life on Our Planet, have brought the scale of extinction into living rooms worldwide. Social media has amplified conservation campaigns and connected people to wildlife in unprecedented ways. Younger generations, in particular, have shown strong support for environmental protection and species conservation. The shifting public opinion created political space for bold conservation initiatives like the Genetic Heritage Act. Politicians who might have dismissed de-extinction as science fiction a decade ago now saw it as a visionary approach to addressing ecological loss. The law wasn't just a scientific or ethical statement. It was a political one, a recognition that the public cared deeply about extinction and wanted action. This political dimension was crucial. De-extinction requires sustained funding, public support, and long-term commitment. Without political will, even the most scientifically sound revival efforts would fail. The Australian law demonstrated that political will existed, at least in one country. It remained to be seen whether other nations would follow suit. But the law also faced significant challenges. Implementation would require substantial funding for research, habitat restoration, monitoring, and community engagement. The Australian government had committed initial funding, but long-term financial sustainability was uncertain. Scientific hurdles remained formidable. Creating a viable Thylacine embryo was one thing. Gestating it to term in a surrogate mother was another. Raising a healthy individual that could survive in the wild was yet another. Establishing a self-sustaining population would be the ultimate and the most difficult challenge of all. Ethical questions would continue to evolve as the science advanced. What rights would revived species have? How would we ensure their welfare? What would happen if revival efforts failed or caused unintended harm? The law provided a framework for addressing these questions, but it couldn't answer them in advance. Each de-extinction proposal would require careful, case-by-case evaluation. The Oversight Committee would face difficult decisions. Public debate would continue. Scientific understanding would evolve. The law was a beginning, not an end. It opened the door to de-extinction but didn't guarantee success. It created possibilities but also responsibilities. The world was watching. Australia had taken the first step. Now the real work would begin. When the Genetic Heritage Act 2024 received royal assent on September 1, 2024, it marked more than a legislative milestone. It represented a fundamental shift in humanity's relationship with extinction, a recognition that the boundary between life and death, once considered absolute, had become permeable through scientific advancement. But this permeability came with profound responsibility. The act wasn't a blank check for genetic engineering. It was a carefully calibrated legal instrument designed to navigate the treacherous waters between scientific possibility and ecological wisdom, between technological ambition and ethical restraint. To understand its significance, we must examine its provisions not as abstract legal text, but as practical responses to concrete challenges that had emerged from decades of scientific research, ethical debate, and conservation failure. This wasn't legislation born in a vacuum. It was forged in the crucible of Australia's ecological crisis, shaped by indigenous wisdom, informed by international scientific consensus, and tempered by hard lessons from past conservation mistakes. The Act's genius and its controversy lay in its specificity. It didn't just say, de-extinction is allowed. It defined exactly what de-extinction meant, who could do it, under what conditions, and with what safeguards. This specificity was both its strength and its limitation. It provided clarity where none had existed before. But it also drew bright lines that some scientists argued were arbitrary, that some conservationists feared were insufficient, and that some ethicists worried were premature. Understanding these tensions is essential to grasping why this law matters, not just for Australia, but for the future of conservation worldwide. The Act's foundational principle was articulated in its preamble, whereas human activity has caused the extinction of numerous species within living memory, whereas advances in genetic science now offer the possibility of reversing some of these losses, whereas such reversal carries significant ecological, ethical, and cultural implications, whereas responsible stewardship requires careful regulation rather than prohibition or laissez-faire innovation, be it therefore enacted. This preamble established the law's moral framework. It acknowledged human culpability for recent extinctions, a crucial distinction from natural extinction events. It recognized scientific capability without celebrating it uncritically. It emphasized responsibility over rights. And it positioned regulation as the middle path between prohibition, which would drive research underground, and unregulated innovation, which could cause ecological harm. This moral framework informed every subsequent provision. The law wasn't neutral. It carried a value judgment that humans have a responsibility to address the damage we've caused, but that this responsibility must be exercised with humility, caution, and respect for ecological complexity. This value judgment would be tested repeatedly in the law's implementation, but it provided a consistent ethical compass for decision-making. Section B of the Act defined the scope of permissible de-extinction with surgical precision. De-extinction was legally defined as the creation of a living organism that possesses genomic characteristics substantially equivalent to those of a species that ceased to exist as a reproducing population within the 50 years preceding the date of this act. This definition contained three critical limitations. First, the 50-year window. As discussed in part B, this wasn't arbitrary. Dead degrades over time, with fragmentation increasing exponentially after death. While advances in synthetic biology might eventually overcome this limitation, the law established a conservative threshold based on current scientific consensus. More importantly, the 50-year window ensured that revived species would have recognizable ecological niches. Ecosystems change over centuries. A species extinct for Bush's years would return to a world it no longer recognized, a world where competitors, predators, prey, and habitats had all shifted. The law recognized that de-extinction without ecological context would be ecologically meaningless and potentially cruel. Second, the requirement for substantially equivalent genomic characteristics. This provision acknowledged that perfect genetic resurrection was impossible. Ancient DNA is always damaged. Some sequences would be missing. Some would be ambiguous. The law permitted scientists to fill these gaps using the genome of the closest living relative, but only where necessary, and only with transparent documentation of all edits. This substantially equivalent standard balanced scientific feasibility with ecological authenticity. A revived thylacine wouldn't be genetically identical to its mammoths ancestors, but it would be close enough to fulfill the same ecological role, exhibit the same behaviors, and carry the same evolutionary heritage. Third, the requirement that the original species cease to exist as a reproducing population. This provision excluded species that still existed in captivity or as isolated individuals. It focused the law on true extinctions, not endangered species that could be saved through traditional conservation. This was crucial for preventing conservation moral hazard, the risk that de-extinction would divert resources and attention from protecting species still clinging to survival. Section 6 established the De-Extinction Oversight Committee, DEOC, the law's central governance mechanism. The committee was mandated to have 11 members, 2 geneticists with expertise in ancient DNA and CRISPR technology, 2 ecologists with experience in species reintroduction and ecosystem restoration, 1 veterinary scientist specializing in wildlife health, 1 ethicist with background in environmental philosophy, 1 legal scholar specializing in environmental law and indigenous rights, 2 representatives from Aboriginal and Torres Strait Islander communities nominated by the National Native Title Council, and 2 public members appointed for their expertise in science communication and community engagement. This composition was deliberate. It ensured that de-extinction decisions wouldn't be made by scientists alone. Ecological, ethical, legal, cultural, and public perspectives would all have formal representation. The committee's powers were extensive but not absolute. It could approve, reject, or conditionally approve de-extinction proposals. It could require additional research, monitoring, or consultation before granting approval. It could revoke approval if conditions were violated or if new evidence emerged suggesting harm. But it couldn't initiate de-extinction projects itself. That power remained with research institutions, which had to submit detailed proposals for committee review. The approval process was designed to be rigorous, but not prohibitive. Proposals had to include a complete genomic analysis demonstrating feasibility, an ecological assessment identifying the species' historical role and current niche availability, a reintroduction plan with habitat restoration components, an animal welfare protocol for gestation, birth, and rearing, a long-term monitoring framework, a consultation record with affected indigenous communities, and a funding plan demonstrating financial sustainability for at least 20 years. This comprehensive requirement ensured that only well-prepared, well-resourced projects would proceed. It filtered out speculative or underfunded efforts that might fail and cause harm to revived species or ecosystems. Section 1 could contain the law's most controversial provisions, the restrictions designed to prevent abuse and ensure ecological responsibility. Subsection 1 a prohibited commercial exploitation of revived species or their genetic material. No patents on de-extinct genomes. No sale of revived animals or their offspring. No use in entertainment, advertising, or private collections. This provision was fiercely debated during legislative hearings. Biotechnology companies argued that commercial incentives were necessary to attract private investment for expensive de-extinction research. They warned that without patent protection, companies would have no incentive to develop the technologies required. Conservation groups countered that commercialization would inevitably lead to exploitation, revived species treated as commodities rather than ecological agents. Genetic material hoarded rather than shared, revival efforts skewed toward charismatic species that could generate revenue rather than ecologically important ones. The law ultimately sided with conservation concerns, establishing de-extinction as a public good rather than a private enterprise. However, it included a compromise. Research institutions could recover reasonable costs associated with de-extinction projects through licensing agreements for non-commercial research or educational purposes. This allowed universities and nonprofits to sustain their work without creating profit motives that could distort ecological priorities. Subsection 1 required that all revived species be released into protected wild habitats, not kept in captivity except for temporary quarantine or medical treatment. This provision reflected the law's ecological purpose. De-extinction wasn't about creating zoo exhibits or private collections. It was about restoring ecological functions. A Thylacine in a zoo wouldn't control feral cat populations. A passenger pigeon in an aviary wouldn't disperse forest seeds. The law recognized that true de-extinction required rewilding, returning species to their ecological roles in functioning ecosystems. This requirement posed significant challenges. Suitable protected habitats had to be identified, secured, and restored. Threats like invasive species, habitat fragmentation, and climate change had to be mitigated. Long-term monitoring and adaptive management plans had to be established. These weren't optional extras. They were legal requirements without which de-extinction approval would be denied. Subsection 1 mandated free, prior, and informed consent from indigenous communities whose traditional lands, cultural heritage, or spiritual beliefs would be affected by revival efforts. This provision was groundbreaking. It recognized that indigenous peoples have unique relationships with extinct species, relationships based on millennia of coexistence, cultural significance, and spiritual connection. For Aboriginal Tasmanians, the Thylacine, known as Lula or Kara, wasn't just an extinct marsupial. It was a totemic being, a cultural symbol, a part of their ancestral heritage. Reviving the Thylacine without their consent would be a form of cultural appropriation, a violation of their rights, and a repetition of colonial patterns of dispossession. The law required that indigenous communities be consulted from the earliest stages of proposal development, that their knowledge and perspectives be incorporated into ecological and cultural assessments, and that they have veto power over projects affecting their traditional lands. This wasn't just ethical. It was practical. Indigenous knowledge of historical species distributions, behaviors, and ecological roles could significantly improve reintroduction success. Indigenous land management practices could provide models for habitat restoration. Indigenous cultural protocols could guide respectful engagement with revived species. The law recognized that de-extinction couldn't succeed without indigenous partnership. Section 1 bis established funding mechanisms and financial safeguards. The Act created the De-Extinction Research and Restoration Fund, capitalized with $2 million from the federal government, and matched by state contributions. The fund would provide grants for approved de-extinction projects, with priority given to projects demonstrating strong ecological benefits, indigenous partnership, and long-term sustainability. But the law also included a critical provision: no project could receive funding without demonstrating a sustainable financial plan for at least 20 years of post-revival monitoring, management, and community engagement. This 20-year rule was designed to prevent the revive and abandon scenario that had plagued some conservation efforts, where initial enthusiasm generated funding for creation, but not for long-term stewardship. The law recognized that de-extinction wasn't a one-time event, it was a multi-generational commitment. A revived Thylacine population would need decades of monitoring, protection, and adaptive management to become self-sustaining. The 20-year rule ensured that this commitment was financially secured before revival began. It also required projects to develop diversified funding strategies, combining government grants, philanthropic support, ecotourism revenue, or appropriate, and community contributions, rather than relying on single sources that might disappear. This financial prudence was essential for building public trust. Taxpayers and donors needed assurance that their money would support lasting conservation outcomes, not scientific spectacles that would fail when funding dried up. Section 1 addressed international obligations and cooperation. Australia is a signatory to the Convention on Biological Diversity, the Convention on International Trade in Endangered Species, CITES, and the Nagoya Protocol on Access and Benefit Sharing. The Act explicitly aligned de-extinction efforts with these international frameworks. Revived species would be treated as endangered under CITES, with strict controls on international movement. Genetic material used in de-extinction would be subject to the Nagoya Protocol's requirements for fair and equitable sharing of benefits arising from genetic resources. The law also established a framework for international collaboration, allowing Australian researchers to partner with foreign institutions while ensuring that all research complied with Australian standards and that all benefits flowed back to conservation efforts in Australia. This international dimension was crucial. De-extinction technology would inevitably spread beyond Australia's borders. By establishing high standards domestically, Australia could influence global norms and prevent a race to the bottom where countries with lax regulations became de-extinction havens. The law positioned Australia not just as a pioneer in de-extinction science, but as a leader in de-extinction governance, a model for responsible innovation that other nations could follow. This leadership role was already bearing fruit. In late 2024, the European Union began drafting its own de-extinction framework, explicitly citing the Australian Act as a reference point. The United States National Academies of Sciences, Engineering, and Medicine launched a study on de-extinction governance, with Australian officials providing testimony. Even China, which had previously pursued genetic engineering with minimal oversight, announced plans to develop ethical guidelines for de-extinction research, citing international pressure and the Australian example. The law was creating ripple effects far beyond Australia's shores. But the act wasn't without its critics. Conservation biologists like Jurassic Park of the Australian National University argued that the law diverted attention and resources from protecting currently endangered species. We're spending millions to revive one extinct species while dozens of living species slide toward extinction every year, she testified during parliamentary hearings. This is conservation theater, not conservation action. Her concerns were legitimate. Australia has over 600 species listed as threatened under the Environment Protection and Biodiversity Conservation Act. Many receive inadequate funding and protection. The de-extinction funds $2 million could have supported habitat restoration, invasive species control, or anti-poaching efforts for multiple endangered species. Proponents countered that de-extinction funding was largely new money, philanthropic donations and private investments attracted specifically by the scientific novelty of revival efforts. They argued that de-extinction could generate public interest and political will for broader conservation efforts. The charisma of revived species like the Thylacine could serve as flagship ambassadors for ecosystem protection, benefiting countless other species sharing the same habitats. This debate reflected a deeper tension in conservation philosophy. Should we focus resources on preventing imminent losses, or should we invest in restoring past losses? The law attempted to bridge this divide by requiring that de-extinction projects demonstrate net positive impact on overall biodiversity, not just the revival of a single species. A Thylacine reintroduction project, for example, would need to show how it would benefit other Tasmanian species through predator control, habitat restoration, or increased conservation funding. This biodiversity multiplier requirement was designed to ensure that de-extinction served broader conservation goals rather than competing with them. Ethicists raised different concerns. Thomas Douglas of Oxford University's Uhuru Center for Practical Ethics questioned whether humans had the right to play God by reviving extinct species. We caused these extinctions through arrogance and ignorance, he wrote in a widely cited commentary. Now we propose to reverse them through further technological intervention. This isn't humility. It's hubris dressed as redemption. His critique resonated with many who view de-extinction as an attempt to escape moral responsibility rather than confront it. If we believe we can simply bring species back later, we might become complacent about preventing extinctions happening today. The law addressed this concern through its emphasis on prevention. Section 2 explicitly stated that de-extinction is not a substitute for preventing extinctions of currently threatened species, and required the government to increase funding for traditional conservation efforts proportionally to de-extinction spending. This no net loss provision ensured that de-extinction wouldn't cannibalize existing conservation budgets. It also mandated public education campaigns highlighting the irreplaceable value of living biodiversity and the importance of preventing extinctions before they occur. These provisions acknowledge the ethical risk of moral hazard while attempting to mitigate it through policy design. Whether they would succeed remained to be seen. Public perception is difficult to control. The media loves charismatic revival stories. Scientists bring back extinct tiger generates more clicks than government funds habitat protection for endangered frog. The law recognized this reality and attempted to counter it through mandatory messaging requirements. All de-extinction projects had to include educational components emphasizing that prevention is always preferable to revival. Indigenous perspectives on the law were complex and varied. Some Aboriginal leaders welcomed the emphasis on consultation and consent. For too long, decisions about our country have been made without us, said Jurassic Park, a Trollope woman, an environmental researcher. This law recognizes that we have knowledge, rights, and responsibilities that must be respected. Others were more cautious. The Thylacine is part of our dreaming, explained Uncle Graham Paul, an Aboriginal elder from Tasmania. Its spirit remains with us. But bringing back its physical form, that's a big decision. We need time to discuss this with our communities, to consult with elders, to understand what this means spiritually and culturally. The law's requirement for free, prior, and informed consent gave Indigenous communities this time. It prevented rushed decisions driven by scientific enthusiasm or political pressure. It recognized that indigenous knowledge systems operate on different timelines than Western science, timelines based on relationship, reciprocity, and intergenerational wisdom rather than publication deadlines and funding cycles. This cultural sensitivity was one of the law's greatest strengths. It acknowledged that de-extinction wasn't just a scientific or ecological issue. It was a cultural and spiritual one. How a society relates to extinct species reflects its values, its history, and its vision for the future. The law created space for these deeper conversations to occur. The practical challenges of implementing the law were immense. Scientific feasibility was just the beginning. Creating a viable Thylacine embryo required overcoming numerous technical hurdles, extracting high-quality DNA from century-old museum specimens, editing the genome of the fat-tailed dunnart, a small marsupial relative, to match Thylacine sequences, developing artificial wombs or surrogate protocols for gestation, ensuring proper development of pouch young, and preparing individuals for life in the wild. Each step carried risks of failure, suffering, or unintended consequences. The law's animal welfare provisions required that all procedures minimize pain and distress, that failed attempts be documented and learned from, and that no individual be subjected to unnecessary suffering. This ethical constraint sometimes slowed progress. Scientists couldn't simply try everything and see what worked. They had to justify each procedure, demonstrate its necessity, and ensure human treatment at every stage. This was frustrating for some researchers who viewed ethical constraints as obstacles to innovation. But it was essential for maintaining public trust and ensuring that de-extinction served conservation rather than scientific curiosity alone. The law recognized that the means matter as much as the ends. How we revive species is as important as whether we revive them. Financial sustainability posed another challenge. The 20-year funding requirement was ambitious. Conservation projects often struggle to secure long-term funding. Political priorities shift. Donor interests change. Economic downturns reduce available resources. The law required projects to develop diversified, resilient funding strategies, but it couldn't guarantee their success. Some proposed solutions included endowment funds invested to generate perpetual income, ecotourism revenue sharing agreements with local communities, corporate sponsorship with strict ethical guidelines, and government commitments locked in through legislation. Each option had trade-offs. Endowments required large initial capital. Ecotourism risked disturbing wildlife. Corporate sponsorship raised concerns about influence. Government commitments were vulnerable to political change. The law didn't prescribe a single solution. It required projects to demonstrate financial viability through whatever combination of strategies made sense for their context. This flexibility was necessary but also created uncertainty. Would revived species become financial burdens when initial enthusiasm faded? The law attempted to mitigate this risk through mandatory contingency planning, requiring projects to identify alternative funding sources, develop phased reduction strategies, and establish emergency protocols for transferring stewardship if primary funders withdrew. These provisions acknowledged that financial sustainability was as critical as scientific feasibility for long-term success. International reach into the law continued to evolve throughout late 2014 and early 2015. Conservation organizations generally praised its cautious approach. The World Wildlife Fund issued a statement calling it a thoughtful framework that balances innovation with responsibility. The International Union for Conservation of Nature, IUCN, incorporated elements of the Australian model into its updated guidelines for reintroductions and other conservation translocations. But some scientists criticized the 50-year rule as unnecessarily restrictive. We have high-quality DNA from woolly mammoths preserved in permafrost, argued George Church of Harvard University, a pioneer in de-extinction research. The ecological argument for reviving mammoths, to restore the mammoth steppe ecosystem and combat permafrost, though, is compelling. Why should arbitrary time limits prevent us from addressing climate change? His critique highlighted a tension within the de-extinction community, between those focused on recently extinct species with clear ecological roles, and those pursuing more ambitious projects with broader climate or ecological benefits. The Australian law represented the former approach. It prioritized ecological realism over technological possibility. It recognized that successful de-extinction required not just scientific capability, but ecological context, social acceptance, and long-term commitment. Projects lacking these elements, even if scientifically feasible, were unlikely to succeed. The law's restrictions weren't about limiting science. They were about ensuring that science served conservation rather than the reverse. As January 2025 approached, the date when the Genetic Heritage Act would take full effect, the world watched closely. Would the first de-extinction proposals meet the law's rigorous standards? Would the De-Extinction Oversight Committee approve any projects in its first year? Would indigenous communities grant consent for Thylacine revival? Would funding materialize for long-term stewardship? These questions had no easy answers. The law had created a framework for responsible de-extinction, but it couldn't guarantee success. It had established guardrails, but the journey ahead remained uncertain. What was certain was that the conversation had changed. De-extinction was no longer a theoretical debate or a science fiction fantasy. It was a legal reality with rules, responsibilities, and real-world consequences. The Australian law had transformed de-extinction from a question of can we to a question of should we, and if so, how. This shift in framing was profound. It moved the discussion from technological possibility to ethical responsibility, from scientific curiosity to ecological purpose, from individual ambition to collective stewardship. The law didn't provide all the answers, but it created the conditions for asking better questions. And in an era of unprecedented ecological loss, better questions might be the most valuable contribution of all. The Genetic Heritage Act wasn't the end of the de-extinction debate. It was the beginning of a new chapter, one where science, ethics, ecology, and culture would have to work together to navigate the uncharted territory of reversing extinction. The world was watching. Australia had set the standard. Now it was up to all of us to rise to the challenge. In a sterile laboratory at the University of Melbourne's TIGRR Lab, Thylacine integrated genomic restoration research, a team of scientists huddled around a microscope, watching as a single cell divided for the first time. This wasn't just any cell. It was a Thylacine embryo created not through natural reproduction, but through the precise editing of marsupial DNA using CRISPR-Cas9's gene editing technology. For the first time in 88 years, the genetic blueprint of the Tasmanian tiger was alive again. But this wasn't magic. It wasn't science fiction. It was the culmination of decades of scientific progress in insane DNA extraction, comparative genomics, and precision gene editing technologies that had advanced so rapidly that what seemed impossible a generation ago had become routine laboratory practice. The resurrection of the Thylacine wasn't about cloning a perfect replica of the extinct animal. It was about editing the genome of its closest living relative, the fat-tailed dunnart, to match the Thylacine's genetic sequence at key locations that determine physical characteristics, behaviors, and ecological functions. This approach, called genetic rescue rather than true de-extinction, acknowledged the impossibility of perfect resurrection while pursuing the practical goal of restoring an ecological role. The science was complex, but the principle was simple: find the genetic differences between the extinct species and its living relative, and edit those differences back into the living genome. The result wouldn't be genetically identical to a Thylacine, but it would be close enough to fulfill the same ecological niche, exhibit the same behaviors, and carry the same evolutionary heritage. This wasn't about creating museum specimens. It was about restoring ecosystem function. And the science was finally ready. The journey began with DNA extraction from museum specimens. The last known Thylacine, Benjamin, died at Hobart Zoo on September 7, 1928. His body was preserved as a taxidermy mount, and tissue samples were stored in ethanol at the Tasmanian Museum and Art Gallery. These samples, along with others from Thylacines that died in the decades before extinction, became the foundation for genomic research. But extracting usable DNA from 80-year-old specimens was like trying to read a book that had been soaked in water, left in the sun, and partially eaten by insects. DNA degrades over time through hydrolysis, oxidation, and microbial activity. Even under ideal preservation conditions, DNA has a half-life of approximately 20 years, meaning that after 5 centuries, half the bonds between nucleotides will have broken. After 80 years, significant fragmentation has occurred, but enough intact sequences remain to reconstruct the genome with modern techniques. The challenge wasn't just degradation. It was contamination. Museum specimens are handled by countless people over decades. They're exposed to dust, mold, bacteria, and modern DNA that can infiltrate the samples and confound analyses. Early attempts to sequence Thylacine DNA in the Yikimung yielded mostly human and bacterial sequences, contaminants that had to be painstakingly filtered out. It wasn't until the development of next-generation sequencing technologies and the Yikimung ones that scientists could generate enough sequence data to overcome this noise. These technologies, like Illumina sequencing in Oxford Nanopore, could read millions of DNA fragments simultaneously, generating terabytes of data from a single sample. By comparing these fragments to reference genomes from related marsupials, scientists could identify which sequences belonged to the Thylacine and which were contaminants. This computational filtering was as important as the wood lab work. It required sophisticated bioinformatics pipelines, machine learning algorithms, and massive computing power to assemble the fragments into a coherent genome. The first complete Thylacine genome was published in Ecological, but it was riddled with gaps and uncertainties. It took another 7 years of refinement, using multiple specimens and advanced assembly techniques to produce the high-quality reference genome that would serve as the template for de-extinction. But having a reference genome was only the beginning. The real challenge was understanding what the genome meant, identifying which genes controlled which traits, which regulatory sequences determined when and where genes were expressed, and which genetic variations distinguished the Thylacine from its living relatives. This required comparative genomics, aligning the Thylacine genome with genomes from other marsupials to identify regions of difference. The fat-tailed dunnart, Sminthopsis crassicaudata, emerged as the ideal candidate for comparison. This small, mouse-sized marsupial carnivore is the Thylacine's closest living relative, sharing a common ancestor approximately 1 million years ago. Despite their dramatic differences in size, Thylacines weigh 30, dunnarts weigh 20 grams, their genomes are remarkably similar, approximately 20 identical at the nucleotide level. This similarity was both a blessing and a curse. It meant that dunnart cells could potentially serve as surrogates for Thylacine embryos. But it also meant that identifying the critical piece of differences that made a Thylacine, rather than just a large dunnart, required extraordinary precision. Scientists focused on genes known to control body size, skull shape, dentition, coat patterning, and reproductive biology. They used CRISPR screens to which genetic edits produced which phenotypic changes in dunnart cells. They compared Thylacine fossils and museum specimens to identify morphological differences that could be linked to specific genetic variations. They studied the development of other marsupials to understand how genetic changes during embryogenesis produced adult characteristics. This detective work was painstaking. Each gene edit had to be validated through multiple rounds of testing. Each phenotypic prediction had to be confirmed through comparative anatomy. Each regulatory sequence had to be verified through gene expression studies. The goal wasn't to edit every difference between Thylacine and dunnart genomes, that would be impossible and unnecessary. The goal was to identify the minimal set of edits required to produce an animal that looked, behaved, and functioned like a Thylacine. This minimal edit approach was crucial for practical and ethical reasons. Fewer edits meant higher success rates, lower risk of unintended consequences, and greater public acceptance. It also acknowledged that perfect genetic resurrection was impossible and perhaps undesirable. A revived Thylacine would need to adapt to a modern world that was different from the Tasmania of Montourge's. Some genetic flexibility might actually be beneficial for long-term survival. The actual gene editing process used CRISPR-Cas9 technology, which had revolutionized genetic engineering since its development in ecological. CRISPR, clustered regularly interspaced short palindromic repeats, is a natural defense mechanism used by bacteria to fight viral infections. Scientists harnessed this mechanism to create a programmable gene editing tool that could cut DNA at precise locations and insert new sequences. The Cas9 protein acts as molecular scissors, guided by a shortened sequence that matches the target DNA. When the guide RNA finds its match, Cas9 cuts both strands of the DNA double helix. The cell's natural repair machinery then kicks in, either stitching the cut ends back together, often introducing errors that disable the gene, or incorporating a new DNA template provided by scientists, allowing precise edits. For Thylacine de-extinction, scientists used the latter approach, homology-directed repair, to insert Thylacine-specific sequences into dunnart cells. The process began with dunnart-induced pluripotent stem cells, IPSCs, adult cells that had been reprogrammed to an embryonic-like state, capable of differentiating into any cell type. These IPSCs were ideal for gene editing because they could be grown indefinitely in culture, allowing scientists to have multiple edits before committing to embryo creation. Using CRISPR-Cas9, scientists made thousands of precise edits to the dunnart genome, replacing dunnart sequences with Thylacine sequences at key locations. Each edit was verified through sequencing to ensure accuracy. Off-target effects, unintended edits at similar but incorrect locations, were minimized through careful guide RNA design and high-fidelity Cas9 variants. After all edits were complete, the modified IPSCs were differentiated into primordial germ cells, peaks, the precursors to sperm and eggs. These pegs were then injected into developing dunnart embryos, which were implanted into surrogate mothers. The resulting offspring would carry Thylacine-edated germ cells, allowing them to produce Thylacine-like offspring when bred together. This multi-generational approach was necessary because directly edating a complete embryo would be technically challenging and ethically problematic. By editing stem cells first, scientists could verify the edits before creating living animals. This cautious, step-by-step approach reflected the law's emphasis on animal welfare and scientific rigor. But CRISPR editing was only half the battle. Creating a viable embryo required overcoming the unique challenges of marsupial reproduction. Unlike placental mammals, including humans, marsupials give birth to highly altricial young, tiny, underdeveloped joys that complete their development in the mother's pouch. A newborn thylacine would have weighed less than a gram, and been blind, deaf, and hairless, essentially a fetus that continued to develop externally. This reproductive strategy posed unique challenges for de-extinction. First, the edited cells had to be compatible with marsupial embryogenesis, the complex process of early development that differs significantly from placental mammals. Scientists had to ensure that Thylacine-edited genes would be expressed at the right times, and in the right tissues during this critical period. Second, the surrogate mother, likely a fat-tailed dunnart or a larger marsupial like a quoll, had to be able to carry the hybrid embryo to term and provide appropriate pouch care. Dunnarts are much smaller than thylacines, raising concerns about whether they could successfully gestate and nurture a larger joy. Scientists explored several solutions, using larger surrogate species, developing artificial pouches that could provide the right temperature, humidity, and milk composition, or editing additional genes to ensure the joy's size remained within the surrogate's capacity. Third, the joy would need species-specific care after birth, including the right milk composition, which changes dramatically during pouch development. Thylacine milk would have been different from dunnart milk in fat content, protein composition, and immune factors. Scientists worked to recreate Thylacine milk formula based on genomic analysis of milk protein genes, or to edit the surrogate mother's mammary glands to produce Thylacine-like milk. These reproductive challenges were among the most difficult aspects of de-extinction. They required not just genetic engineering, but developmental biology, reproductive physiology, and animal husbandry expertise. Success would depend on integrating knowledge from multiple disciplines, a troop of collaborative science. The first successful Thylacine embryo was created in March 2024, marking a watershed moment in de-extinction science. The embryo was the result of over a decade of research, thousands of failed attempts, and millions of dollars in funding. It represented not just a scientific achievement, but a proof of concept that the genetic rescue approach could work for marsupials. The embryo was created by injecting Thylacine-edated primordial germ cells into a dunnart blastocyst, early-stage embryo, which was then implanted into a surrogate mother. The pregnancy lasted just 12 days, the normal gestation period for dunnarts, before the tiny joey was born and crawled into the surrogate's pouch. For the first time, scientists could observe Thylacine-like development in real time. The joey was smaller than a jelly bean, pink and hairless, with tiny forelimbs that would eventually develop into the Thylacine's distinctive striped coat. Genetic testing confirmed that the joey carried Thylacine-edated sequences at all target locations. But this was just the beginning. The joey would need to survive pouch development, learn to hunt, and eventually breed with other Thylacine-edated individuals to establish a population. The road ahead was long and uncertain. Many challenges remained, ensuring proper immune system development, preventing genetic disorders from off-target edits, maintaining genetic diversity in a small founder population, and preparing the joey for eventual release into the wild. But the first step had been taken. The Thylacine was no longer just a memory preserved in museum specimens and grainy photographs. It was alive again, in a laboratory, in an embryo, in a tiny joey taking its first breaths in a surrogate's pouch. The science of resurrection had moved from theory to practice. And the world was watching. But the Thylacine wasn't the only candidate for de-extinction. The passenger pigeon, once the most abundant bird in North America, with flocks numbering in the billions, was also on the revival list. Unlike the Thylacine, which had been extinct for less than a century, the passenger pigeon had vanished in 2001 when the last individual, Martha, died at the Cincinnati Zoo. But unlike many extinct species, the passenger pigeon had left behind an abundance of well-preserved specimens in museums across North America. These specimens, combined with advances in avian genomics, made passenger pigeon de-extinction scientifically feasible. The approach was similar to the Thylacine project: edit the genome of the closest living relative, the band-tailed pigeon, to match passenger pigeon sequences at key locations controlling flocking behavior, migratory patterns, and physical characteristics. But birds presented different challenges from marsupials. Avian reproduction involves hard-shelled eggs, rather than live birth, requiring different techniques for embryo manipulation and surrogate incubation. Bird genomes are also more complex, with higher rates of repetitive sequences and chromosomal rearrangements. And passenger pigeon ecology was fundamentally different from Thylacine ecology. The Thylacine was a solitary apex predator. The passenger pigeon was a highly social, nomadic species that depended on massive flocks for survival. Reviving a single passenger pigeon wouldn't be enough. Scientists would need to create hundreds or thousands of individuals simultaneously to recreate the social dynamics essential for the species' survival. This critical mass problem was unique to a highly social species and posed significant logistical and ethical challenges. How many individuals would be needed to establish a viable population? How would they learn species-specific behaviors without adult mentors? How would they navigate migratory routes that had been lost for a century? These questions had no easy answers. But the passenger pigeon project was advancing rapidly, with Harvard's Revive and Restore initiative leading the effort. By late 2022, scientists had successfully edited band-tailed pigeon embryos to express passenger pigeon feather patterns and had begun testing flocking behavior in small groups of edited birds. The goal wasn't just to create passenger pigeon-like birds. It was to restore the ecological function of massive migratory flocks, seed dispersal, forest disturbance, nutrient cycling, that had shaped eastern North American ecosystems for millennia. Like the Thylacine project, the passenger pigeon revival was about more than scientific curiosity. It was about ecological restoration. The science of de-extinction was advancing on multiple fronts simultaneously. While the Thylacine and passenger pigeon projects captured headlines, dozens of other species were being considered for revival under the Genetic Heritage Act's framework. The gastric brooding frog, unique for its ability to incubate eggs in its stomach, was a prime candidate due to its well-preserved DNA and clear ecological role in Australian freshwater ecosystems. The Tristmas island rat, extinct since mammoths, was being studied as a model for rodent de-extinction. Even the woolly mammoth, extinct for thousands of years, was being discussed, though it fell outside the Act's 50-year window and would require different legal frameworks. Each species presented unique scientific challenges and ecological opportunities. The common thread was the recognition that de-extinction wasn't a one-size-fits-all process. Each revival effort required tailored approaches based on the species' biology, ecology, and conservation status. What worked for marsupials might not work for birds. What worked for solitary predators might not work for social herbivores. The science had to be flexible, adaptive, and species-specific. This customization was both a strength and a limitation of de-extinction. It allowed scientists to develop optimal strategies for each candidate species. But it also meant that success in one project didn't guarantee success in another. Each revival effort would require its own research program, funding stream, and regulatory approval. The field was expanding rapidly, but it was also fragmenting into specialized subdisciplines. Geneticists focused on genome editing. Developmental biologists studied embryogenesis. Ecologists planned reintroductions. Ethicists debated implications. This specialization was necessary for scientific progress, but it also created communication challenges. Scientists from different disciplines sometimes struggled to understand each other's work or appreciate each other's constraints. Bridging these gaps required interdisciplinary collaboration, shared language, and mutual respect. The most successful de-extinction projects were those that integrated expertise from multiple fields from the early stages of planning. But the science also faced significant limitations. Despite advances in CRISPR technology, gene editing wasn't perfect. Off-target effects, unintended edits at similar but incorrect locations, remained a concern, particularly for complex traits controlled by multiple genes. Epigenetic factors, chemical modifications to DNA that affect gene expression without changing the underlying sequence, were poorly understood and difficult to replicate. Developmental plasticity, the ability of an organism to adapt its development based on environmental conditions, meant that genetically identical individuals could develop differently depending on their experiences. These limitations meant that revived species would never be perfect replicas of their extinct ancestors. They would be hybrids, part original, part edited, part adapted to modern conditions. This wasn't necessarily a bad thing. Evolution is about adaptation, not preservation. A revived thylacine that could survive in modern Tasmania might need genetic differences from its mammoths ancestors. The key question was whether these differences would compromise the species' ecological function or cultural significance. Would a Thylacine with some Dunner gin still be a Thylacine? Would a passenger pigeon raised by band-tailed pigeons still exhibit the behaviors that made the species unique? These questions had no definitive answers. They required careful monitoring, adaptive management, and ongoing evaluation. The science of de-extinction was still young. It would evolve as researchers learned from successes and failures. Each revived species would provide new insights into genetics, development, and ecology. Each challenge would drive innovation in technology and methodology. The field was entering a period of rapid learning and adaptation, a scientific revolution in real time. The ethical implications of this scientific progress were profound. Every technical advance raised new ethical questions. Was it right to create animals that might suffer due to genetic defects or developmental abnormalities? Was it fair to use surrogate mothers of other species to gestate hybrid embryos? Was it responsible to release genetically edited animals into the world, where they might interact unpredictably with existing ecosystems? These questions couldn't be answered by science alone. They required input from ethicists, indigenous communities, animal welfare experts, and the public. The Genetic Heritage Act recognized this by mandating multidisciplinary review for each de-extinction proposal. But the science was moving faster than the ethics. New capabilities emerged before their implications could be fully understood. Scientists found themselves making decisions with incomplete information, balancing potential benefits against uncertain risks. This tension between scientific possibility and ethical responsibility was at the heart of the de-extinction debate. Some argue that the precautionary principle should prevail. Until we can guarantee no harm, we shouldn't proceed. Others countered that the precautionary principle had failed to prevent the biodiversity crisis in the first place. Sometimes, bold action was necessary to address urgent problems. The middle path was adaptive management: proceed cautiously, monitor closely, adjust based on evidence, and be prepared to intervene if problems arise. This approach acknowledged uncertainty while allowing progress. It recognized that perfect knowledge was impossible, but that didn't mean we should do nothing. The science of de-extinction was inherently uncertain. But so was the alternative: continuing to lose species at an accelerating rate while waiting for perfect solutions. Sometimes, imperfect action was better than perfect inaction. As ecological approached, the science of de-extinction stood at a crossroads. The technologies existed. The legal framework was in place. The first embryos had been created. But the real work was just beginning. Creating a single Thylacine joy was one thing. Establishing a self-sustaining population was another. Reintroducing that population into the world was yet another. Each step would require years of research, monitoring, and adaptation. Each success would be tempered by failures. Each breakthrough would raise new questions. The science was ready. But was society ready? Were we prepared to accept the ethical responsibilities of playing a role in reversing extinction? Were we willing to commit the resources, patience, and humility required for long-term stewardship? These weren't scientific questions. They were human ones. And they would determine whether de-extinction became a transformative conservation tool or a cautionary tale of technological hubris. The Thylacine embryo in the TIGRR Lab was more than a scientific achievement. It was a mirror reflecting our values, our priorities, and our vision for the future. What we did with that embryo, how we nurtured it, protected it, and eventually released it, would say more about us than about the species we were trying to revive. The science had given us the power to resurrect the dead. Now it was up to us to decide whether and how to use that power wisely. The Thylacine was waiting. The question was whether we were ready to welcome it back. In the pre-dawn mist of Tasmania's wilderness, a lone pademelon freezes mid-step, ears twitching. Something is different. The air carries a scent not smelled in 88 years, a predator's musk, sharp and unfamiliar. The pademelon doesn't know the name Thylacinus cynocephalus. It doesn't know the scent belongs to a species declared extinct in Montourge's Ortzalta. But its instincts remember. Its muscles tense. Its heart races. And in that moment, an ecological clock begins ticking backward. The return of the Thylacine isn't just a scientific achievement. It's an ecological experiment on a continental scale, one that will reshape Tasmania's ecosystems in ways both predictable and profoundly unexpected. For decades, conservation biology has operated on a simple principle: protect what remains. But de-extinction forces us to confront a more complex question: can we restore what's been lost? And if we can, should we? The Thylacine's return offers a real world of this question, not in theory, but in practice. It forces us to examine not just whether we can revive an extinct species, but whether we should, and what responsibilities come with playing a role in reversing extinction. The answers aren't simple. Ecosystems are complex adaptive systems, not clockwork mechanisms. Introducing a top predator after nearly a century of absence will trigger cascading effects, some beneficial, some harmful, many unpredictable. But this uncertainty isn't a reason to avoid action. It's a reason to proceed with humility, rigor, and adaptive management. The Thylacine's return isn't about recreating the past. It's about healing the present. And understanding the ecological ripple effects is essential to doing that wisely. To understand what the Thylacine's return means, we must first understand what its absence has cost. The Thylacine was Tasmania's apex predator, the undisputed ruler of its ecosystem. Weighing 1 kilograms with powerful jaws capable of crushing bone, and a hunting style that combined endurance running with ambush tactics, the Thylacine occupied a unique ecological niche. It preyed primarily on medium-sized marsupials like wallabies, pademelons, and possums, but also took birds, reptiles, and occasionally livestock. Its presence shaped the behavior, distribution, and population dynamics of countless species. Pre-animals evolved vigilance behaviors, freezing in unfamiliar scents, avoiding open areas at dusk, forming tighter social groups. Competitor species like the Tasmanian devil and spotted-tailed quoll adjusted their hunting territories and activity patterns to avoid direct conflict. Even vegetation structure was influenced by the Thylacine's presence, through its effects on herbivore populations that browsed young plants. This is the essence of a trophic cascade. A top predator's influence ripples down through multiple levels of the food web, shaping everything from animal behavior to plant communities to soil chemistry. When the Thylacine vanished, this cascade reversed. Without predation pressure, herbivore populations expanded. Without competition from a larger predator, mesopredators like feral cats and red foxes, introduced to Tasmania in the 19th century, found new opportunities. Without the Thylacine's territorial presence, ecosystems lost a stabilizing force that had maintained balance for millennia. The consequences weren't immediate or dramatic, they unfolded gradually over decades, like a slow-motion ecological unraveling. But by the early 20th century, Tasmania's ecosystem showed clear signs of what ecologists call mesopredator release and trophic downgrading, the degradation of ecosystem function following the loss of top predators. The evidence of this unraveling is everywhere in modern Tasmania. Feral cat populations have exploded, with densities reaching up to cats per square kilometer in some areas, among the highest in the world. These cats are devastating for native wildlife. A single feral cat can kill over native animals per year, small mammals, birds, reptiles that evolved without defenses against this efficient predator. The Tasmanian bettong, eastern barred bandicoot, and swift parrot are just a few of the species pushed to the brink by cat predation. Red foxes, the less established than cats, pose an even greater threat. Their larger size and pack hunting behavior make them capable of taking prey as large as wallabies. Fox eradication programs have cost Tasmania over $2 million since with mixed success. Meanwhile, herbivore populations have surged in the absence of Thylacine predation. Bennett's wallabies now reach densities of 1 individuals per hectare in some areas, levels that cause significant overgrazing, soil erosion, and reduced regeneration of native vegetation. This overgrazing has cascading effects. Fewer young trees mean less habitat for arboreal species like possums and gliders. Compacted soil reduces water infiltration and increases runoff. Simplified vegetation structure supports fewer insect species, which in turn affects birds and reptiles. The loss of the Thylacine didn't just remove one species. It removed a regulatory force that had maintained ecosystem balance for thousands of years. Tasmania's ecosystems didn't collapse, they adapted. But they adapted to a degraded state, one with fewer species, simpler food webs, and reduced resilience to disturbances like fire, disease, and climate change. This degraded state is what scientists call an alternative stable state, a new equilibrium that's less biodiverse and less functional than the original, but stable enough to persist without intervention. Restoring the Thylacine isn't about returning to some pristine past. It's about shifting the ecosystem back toward a more functional, resilient state, one where native species can thrive without constant human intervention. But reintroducing a top predator after nearly a century of absence is not without risks. The most immediate concern is human-wildlife conflict. The Thylacine was eradicated in large part because European settlers viewed it as a threat to livestock, particularly sheep. Historical records show that Thylacines did occasionally take sheep, especially lambs, though studies suggest this was less common than settlers claimed. Many Thylacine attacks were likely misattributed to feral dogs or other predators. Nevertheless, the perception of the Thylacine as a livestock predator was powerful enough to justify government-sponsored bounties that paid hunters for each Thylacine killed. Between 1966 and 1912, the Tasmanian government paid bounties for ecological Thylacines, though the actual number killed was likely much higher. This history looms large in the minds of modern Tasmanian farmers. Would they accept the return of a predator that once threatened their livelihoods? The answer depends on several factors. First, modern livestock management is vastly different from 19th century practice. Electric fencing, Gordian animals, like Marama sheepdogs, nighttime penning, and improved herd monitoring can significantly reduce predation risk. Second, compensation programs can offset any losses that do occur. New South Wales' successful dingo management program, which compensates farmers for verified livestock losses while maintaining dingo populations for ecological benefits, offers a potential model. Third, education and engagement are critical. Farmers need to understand that the Thylacine's ecological role, controlling feral cats and foxes that also prey on lambs, could actually benefit livestock operations in the long run. A ecological study by the University of Tasmania found that areas with healthy dingo populations had fewer feral cat sightings and higher lamb survival rates compared to areas where dingoes were suppressed. The Thylacine could provide similar benefits. But these benefits won't be automatic. They'll require careful planning, transparent communication, and genuine partnership with the agricultural community. The Thylacine's return can't succeed without farmer support. And earning that support will require addressing legitimate concerns while demonstrating tangible benefits. Another risk is disease transmission. 88 years is a long time in the world of pathogens. The Thylacine vanished before the arrival of devil facial tumor disease, DFT, a contagious cancer that has decimated Tasmanian devil populations since the mid-1920s. It vanished before the spread of toxoplasmosis, a parasitic disease carried by feral cats that infects native wildlife. It vanished before the emergence of amphibian chytrid fungus and other modern wildlife diseases. A revived Thylacine would have no evolutionary immunity to these modern pathogens. Conversely, the Thylacine could carry ancient pathogens, or novel ones created through the de-extinction process, that could threaten existing wildlife. This isn't just theoretical. The reintroduction of black-footed ferrets in North America was nearly derailed by sylvatic plague, a disease the ferrets had no immunity to. The release of captive-bred California condors was complicated by lead poisoning from ammunition and microtrash ingestion. Disease risk is one of the most significant challenges in any reintroduction program, and the Thylacine's case is no exception. The Genetic Heritage Act addresses this through rigorous health screening protocols. Every revived Thylacine must undergo comprehensive veterinary examination, pathogen screening, and quarantine before release. They must be vaccinated against known wildlife diseases where vaccines exist. They must be monitored for at least 2 years post-release for signs of illness or unusual behavior. But screening can't eliminate all risks. Some pathogens are difficult to detect. Some have long incubation periods. Some emerge unexpectedly. This is where adaptive management becomes critical. The reintroduction plan includes contingency protocols for disease outbreaks, rapid response teams, emergency capture capabilities, treatment facilities, and communication plans with wildlife health authorities. The goal isn't to eliminate all risk, that's impossible. The goal is to minimize risk through careful preparation while maintaining the flexibility to respond quickly if problems arise. This balance between precaution and action is at the heart of responsible reintroduction science. Perhaps the most profound uncertainty is how the Thylacine itself will adapt to a world that has changed dramatically since 1920. Tasmania in 1920 is not Tasmania in 1920. The climate is warmer and drier in many regions. Rainfall patterns have shifted. Fire regimes have intensified. Human infrastructure, roads, towns, farms has fragmented the landscape. Invasive species like cats, foxes, and weeds are now entrenched. The Thylacine's historical prey species have changed their behaviors and distributions in response to these pressures. Will a revived Thylacine recognize modern paedomolons as prey? Will it avoid roads and vehicles? Will it compete effectively with feral cats for food and territory? Will it find suitable denning sites in a landscape altered by logging and development? These aren't rhetorical questions. They're practical challenges that will determine whether reintroduction succeeds or fails. The Thylacine's genome has been edited to match historical sequences, but behavior isn't encoded solely in DNA. Much of what made a thylacine hunting techniques, social structure, territorial behavior was learned from parents and conspecifics. A revived thylacine raised by fat-tailed dunnerts or human caretakers won't have access to this cultural knowledge. It will need to learn survival skills through trial and error, a process that could be costly in terms of individual mortality. This knowledge gap is one of the most significant challenges in de-extinction. Scientists are addressing it through innovative training programs. Before release, thylacines will undergo predator school enclosures that simulate natural hunting scenarios with live prey, initially captive bred, then will caught under permit. They'll be exposed to recorded sounds of vehicles, humans, and other threats to develop appropriate avoidance behaviors. They'll be fitted with EEPS collars and camera traps to monitor their movements, hunting success, and interactions with other species. This monitoring won't just protect the thylacines. It will generate invaluable data about how revived species adapt to modern ecosystems, data that will inform future de-extinction efforts worldwide. The thylacines' return isn't just about restoring one species. It's about learning how to restore ecological relationships in a changed world. And that learning process will be as important as the restoration itself. The reintroduction plan itself is a masterpiece of ecological engineering, phased, cautious, and adaptive. Phase 2027 involves establishing a captive breeding population of at least 50 genetically diverse thylacines at a secure facility in Tasmania. These individuals will be the founders of the world population, so genetic diversity is critical to avoid inbreeding depression and maintain adaptive potential. Scientists will use genomic analysis to select breeding pairs that maximize genetic variation while minimizing the risk of deleterious mutations from the editing process. Phase 2027 involves soft release trails in large, fenced reserves, areas of hectares where thylacines can hunt, establish territories, and develop natural behaviors without the risks of full wilderness release. These reserves will be stocked with native prey species and monitored intensively. Scientists will study hunting success rates, territorial behavior, social interactions, and health indicators. They'll refine release protocols based on what they learn. Phase 2027 involves the first world releases, initially just 5 to 10 individuals in a carefully selected area of western Tasmania with low human density, high prey-availability, and minimal invasive predator pressure. These pioneers will be monitored via satellite collars, camera traps, and drone surveys. Their survival, reproduction, and ecological impacts will be documented in real time. If they thrive, additional releases will follow in subsequent years. If problems arise, disease outbreaks, human conflicts, ecological disruptions, the program can be paused or modified. This phased approach reflects the precautionary principle in action. It acknowledges uncertainty while allowing progress. It builds in multiple off-ramps if things go wrong. And it generates data at every step to inform the next decision. This isn't reckless experimentation. It's responsible science. The ecological monitoring framework is equally sophisticated. Scientists won't just track the thylacines. They'll track the entire ecosystem's response. Camera traps will document changes in prey behavior and mesopredator activity. Vegetation surveys will measure shifts in plant communities and regeneration rates. Soil samples will analyze changes in nutrient cycling and microbial communities. Acoustic monitors will record changes in bird and insect populations. Citizen science programs will engage local communities in data collection, reporting sightings, tracking prey populations, monitoring vegetation changes. This comprehensive monitoring will answer critical questions: are feral cat populations declining in areas with thylacines? Are native herbivore populations stabilizing or declining? Is vegetation structure becoming more complex? Are other native predators like devils and quolls benefiting from reduced competition? The data will be publicly available through an online dashboard, allowing scientists, policymakers, and the public to track progress in real time. Transparency is essential for building trust and ensuring accountability. If the thylacines' return causes unexpected harm, if it preys heavily on endangered species, for example, the program can be adjusted or halted. Adaptive management isn't just a buzzword. It's a commitment to learning, adjusting, and prioritizing ecosystem health over ideological commitments. The goal isn't to prove that de-extinction works. The goal is to restore ecological function in the most effective, responsible way possible. If that means modifying the approach or even stopping it, the science must guide the decision. Lessons from other reintroduction programs offer both hope and caution. The reintroduction of gray wolves to Yellowstone National Park in Mungtoorustuksan is often cited as a success story, and for good reason. Wolves reduced overabundant elk populations, which allowed willow and aspen to regenerate along riverbanks. This, in turn, created habitat for beavers, songbirds, and fish. River morphology changed as vegetation stabilized banks. Biodiversity increased across multiple trophic levels. This trophic cascade demonstrated the profound influence of apex predators on ecosystem function. But Yellowstone's success wasn't automatic. It required decades of planning, stakeholder engagement, adaptive management, and conflict mitigation. Ranchers were compensated for livestock losses. Hunters were educated about the ecological benefits of wolves. Scientists monitored the ecosystem intensively and adjusted management strategies based on what they learned. The program faced setbacks, disease outbreaks, illegal killings, political opposition, but it persisted because the ecological benefits were clear and the management was flexible. Closer to home, the reintroduction of the eastern quoll to mainland Australia after an 16-year absence offers valuable lessons. Quolls were released into fenced reserves and monitored closely. Some individuals struggled to adapt, avoiding natural prey, failing to establish territories, succumbing to disease. But others thrived, and the population gradually stabilized. Scientists learned that pre-release training was critical. Quolls that practiced hunting live pre before release had significantly higher survival rates. They also learned that genetic diversity mattered. Populations founded by individuals from multiple source populations were more resilient. These lessons are being applied directly to the Thylacine program. But not all reintroductions succeed. The attempt to reintroduce the Arabian oryx to Oman and the Mungtoorustuksan initially succeeded, but poaching and habitat loss eventually caused the population to collapse. The release of captive bred golden lion tamarins in Brazil has required decades of habitat restoration, corridor creation, and community engagement to achieve modest success. These failures aren't reasons to avoid reintroduction. They're reasons to do it better, to learn from past mistakes, to plan more thoroughly, to engage stakeholders more deeply, to monitor more rigorously. The Thylacine program is benefiting from this collective wisdom. It's not reinventing the wheel. It's building on decades of hard-won experience. Indigenous knowledge offers another critical perspective, one that Western science has too often overlooked. For Aboriginal Tasmanians, the Thylacine, Lula or Kara, was never just a predator. It was a totemic being, a cultural symbol, a part of their ancestral heritage. Its disappearance wasn't just an ecological loss. It was a spiritual wound. But Indigenous perspectives on the Thylacine's return are complex and varied. Some elders welcome the opportunity to restore a cultural connection that was severed by colonization. The Lula is part of our dreaming, explains Auntie Patsy Cameron, a Paikana woman and cultural heritage advisor. Its return would heal a part of our country that has been missing for generations. Others are more cautious. We need to ask, is this the right time? Are we ready? Does the land want the Lula back? These questions reflect a worldview that sees humans as part of nature, not separate from it, a worldview that emphasizes reciprocity, responsibility, and long-term thinking. Indigenous knowledge also offers practical insights that Western science lacks. Aboriginal oral histories describe Thylacine behavior, habitat preferences, and seasonal movements that aren't recorded in scientific literature. Traditional ecological knowledge identifies areas where Thylacines were historically abundant, areas that might be suitable for reintroduction today. Indigenous land management practice, like cultural burning, could help prepare habitats for Thylacine return by reducing fuel loads, promoting diverse vegetation, and creating mosaic landscapes that support both predators and prey. The Genetic Heritage Act mandates Indigenous consultation and consent, but the Thylacine program is going further. Aboriginal rangers are being trained as Thylacine monitors. Indigenous knowledge holders are serving on the De-Extinction Oversight Committee. Cultural protocols are being developed to guide respectful engagement with the revived species. This partnership isn't just ethical. It's practical. Indigenous knowledge complements Western science, filling gaps that data alone can't address. Together, they create a more holistic, culturally grounded approach to ecological restoration. The Thylacine's return isn't just a scientific project. It's a cultural healing process, one that acknowledges past wrongs and builds a more inclusive future. Measuring success is perhaps the most challenging aspect of the entire endeavor. What does success even mean for a de-extinction project? Is it simply establishing a self-sustaining population of Thylacines? That's a necessary condition, but not sufficient. A population of 50 thylacines surviving in a fenced reserve isn't ecological restoration. It's conservation captivity. True success requires the thylacine to fulfill its ecological role, to regulate pre-populations, suppress misoppredators, and contribute to ecosystem function. But how do we measure that? Scientists have developed a suite of metrics: thylacine population growth rate and genetic diversity, changes in feral cat and fox activity, shifts in native herbivore behavior and density, vegetation regeneration rates, biodiversity indices across multiple taxa. These metrics will be tracked over decades, not years, to capture long-term trends. But ecological restoration isn't just about numbers. It's about relationships. It's about whether the thylacine becomes re-embedded in Tasmania's ecological and cultural fabric. Does it inspire greed or conservation efforts? Does it reconnect people to nature? Does it heal the spiritual wound of its absence? These qualitative dimensions matter as much as quantitative ones. Success will be measured not just in scientific papers, but in community stories, cultural revitalization, and a renewed sense of hope. The thylacine's return isn't a finish line. It's a beginning, a catalyst for broader ecological and cultural healing. And that healing will take generations to fully realize. As the first thylacines prepare for release into the Tasmanian wilderness, the world watches with bated breath. This isn't just about one species. It's about whether humanity can learn from its mistakes, take responsibility for its actions, and work to heal the damage we've caused. The thylacine's return is not of our technological prowess, but of our wisdom, humility, and commitment to stewardship. It forces us to confront difficult questions. What is our relationship with nature? What responsibilities come with power? What does it mean to live in right relationship with other species? These aren't scientific questions. They're human ones. And the answers will shape not just Tasmania's future, but our own. The thylacine is waiting. The ecosystem is waiting. The question is whether we're ready, not just scientifically, but ethically, culturally, and spiritually, to welcome it back. The ecological ripple has begun. Where it lives depends on us. The question isn't whether we can resurrect the thylacine. The embryo in the TIGRR Lab has already answered that. The question is whether we should, and this question has haunted de-extinction science since its inception. It's a question that divides conservationists, ethicists, indigenous communities, and the public. It's a question with no easy answers, only difficult trade-offs and competing values. At its core, the ethical debate about de-extinction isn't really about technology. It's about responsibility. It's about whether humanity's role in nature is to dominate, to preserve, to restore, or to let go. It's about whether we have the right to reverse decisions made by previous generations, or whether doing so absolves us of the responsibility to prevent extinctions happening today. It's about humility versus hubris, redemption versus arrogance, hope versus despair. These aren't abstract philosophical questions. They're practical ones that will determine whether de-extinction becomes a transformative conservation tool or a cautionary tale of technological overreach. And they require us to confront uncomfortable truths about our relationship with nature, not as distant observers, but as active participants who have already reshaped the planet beyond recognition. The strongest ethical argument for de-extinction is rooted in responsibility. Humans caused the thylacine's extinction. Not natural climate change. Not asteroid impacts. Not evolutionary competition. Humans, European settlers armed with guns, bounties, and a worldview that saw predators as vermin, systematically eradicated the species over decades. This wasn't an accident. It was a deliberate act of ecological violence justified by economic interests and cultural prejudice. The thylacine's extinction represents a moral failure, a failure to recognize the intrinsic value of other species, to respect ecological relationships, to exercise restraint in the face of abundance. If humans bear responsibility for causing extinctions, the argument goes, we also bear responsibility for reversing them when possible. This isn't about playing God. It's about taking responsibility for our actions. It's about acknowledging that we broke something, and now have the tools to fix it. Philosopher Claire Palmer of Texas and University frames this as a duty of repair. When we cause harm through our actions, we have a moral obligation to mitigate that harm when feasible. De-extinction, in this view, isn't hubris. It's humility, the recognition that we made a mistake and now have the opportunity to make amends. This argument resonates particularly strongly in Australia, where the thylacine's extinction is widely recognized as a national shame. For many Australians, reviving the thylacine isn't just ecological restoration. It's atonement, a symbolic act of reconciliation with the natural world and with Aboriginal peoples whose relationship with the species was severed by colonization. This moral dimension can't be dismissed as sentimental. It reflects a deeper truth: conservation isn't just about preserving biodiversity. It's about healing relationships between humans and nature, between past and present, between colonizers and indigenous peoples. De-extinction, when approached with humility and respect, can be part of that healing process. But the counterargument is equally compelling. De-extinction risks creating conservation moral hazard, the dangerous belief that extinction is reversible, and therefore less urgent to prevent. If people believe we can simply bring species back later, they may become complacent about habitat destruction, climate change, and other threats driving the current extinction crisis. Why protect rainforests today if we can resurrect orangutans tomorrow? Why reduce carbon emissions if we can engineer climate-resilient species? This isn't hypothetical. Psychological studies have shown that the mere perception of technological solutions reduces willingness to make behavioral changes. In a ecological experiment published in Nature Climate Change, participants who were told about carbon capture technology subsequently expressed less support for emissions reductions than those who weren't. The techno-fix mentality is seductive because it promises solutions without sacrifice. De-extinction risks becoming the ultimate techno-fix, a spectacle that distracts from the hard, unglamorous work of habitat protection, pollution reduction, and sustainable development. Conservation biologist Stuart Pym of Duke University warns, we're losing species at 1,000 times the natural rate. De-extinction might bring back one or two. It won't solve the biodiversity crisis. In fact, it might make it worse by diverting attention and resources from prevention. His concern is practical, not philosophical. Australia has over 100 species listed as threatened. The odd $2 million de-extinction fund could protect habitat for dozens of these species, but it's being spent to revive one extinct one. This isn't an either-or choice. De-extinction funding is largely new money attracted by scientific novelty, but the opportunity cost is real. Every dollar spent on revival is a dollar not spent on prevention. Every headline about Thylacine embryos is a headline not about critically endangered frogs. The ethical question isn't just, should we revive extinct species? It's at what cost to living ones. And until we can answer that honestly, de-extinction remains ethically ambiguous. The animal welfare dimension adds another layer of complexity. Is it ethical to create an animal that may suffer due to genetic defects, developmental abnormalities, or inability to thrive in a changed world? CRISPR editing isn't perfect. Off-target effects, unintended edits at similar but incorrect locations, can cause health problems ranging from minor anomalies to lethal disorders. Epigenetic factors, chemical modifications affecting gene expression, can't be fully replicated from century-old DNA. Developmental plasticity means that genetically identical individuals raised in different environments develop differently. A thylacine raised by fat-tailed dunards or human caretakers won't learn species-specific behaviors from parents. It may struggle to hunt, avoid predators, or interact appropriately with conspecifics. It may experience psychological distress from isolation or inappropriate socialization. These aren't hypothetical concerns. Cloning and genetic engineering have high failure rates. Dolly the sheep was the only success out of ecological attempts. Many cloned animals suffer from premature aging, organ failure, or immune dysfunction. The Thylacine program has already experienced losses, embryos that failed to develop, joys that died shortly after birth, individuals with developmental abnormalities that required euthanasia. Each loss represents a sentient being that suffered for human ambition. Is this suffering justified by the goal of species revival? Animal ethicist Peter Singer argues that the moral stats of an animal depends on its capacity to suffer, not its species identity or conservation value. From this perspective, creating animals likely to suffer for human purposes, even noble ones like conservation, is ethically problematic unless the suffering is minimized and the benefits are substantial. The Genetic Heritage Act attempts to address this through rigorous animal welfare protocols, minimizing the number of experimental subjects, using non-invasive monitoring techniques, providing enriched environments, and euthanizing individuals experiencing unrelievable suffering. But these protocols can't eliminate suffering entirely. They can only reduce it to acceptable levels, a judgment that itself raises ethical questions. Who decides what level of suffering is acceptable? How do we weigh the suffering of individual animals against the potential benefits to ecosystems and future generations? These aren't questions science can answer alone. They require ethical deliberation, public dialogue, and humility in the face of uncertainty. Indigenous perspectives complicate the ethical landscape further, often in ways Western frameworks fail to anticipate. For many Aboriginal Australians, the Thylacine, Lula, isn't just an extinct marsupial. It's a totemic being with spiritual significance, a part of their ancestral heritage, a thread in the fabric of their cultural identity. Its disappearance wasn't just an ecological loss. It was a spiritual one inflicted by colonization. But Indigenous views on de-extinction aren't monolithic. Some elders welcome the opportunity to restore a severed cultural connection. The Lula is part of our dreaming, explains Auntie Patsy Cameron. Its return would heal a part of our country that has been missing for generations. Others express deep reservations. Uncle Graham Paul, an Aboriginal elder from Tasmania, asks, "Is this the right time? Are we ready? Does the land want the Lula back?" These questions reflect a worldview fundamentally different from Western conservation paradigms, one that sees humans as part of nature rather than separate from it, that emphasizes reciprocity rather than domination, that values spiritual relationships alongside ecological ones. Some Indigenous knowledge holders worry that de-extinction represents another form of colonial appropriation, taking genetic material from ancestral beings without proper ceremony, consent, or reciprocity. They ask, who owns the Thylacine? Who has the right to play with ancestral spirits? What obligations come with reviving a being that was deliberately killed by colonizers? These aren't objections to science per se. They're demands for respect, for recognition that Indigenous peoples have unique relationships with extinct species that must be honored through proper protocols, consultation, and consent. The Genetic Heritage Act's requirement for free, prior, and informed consent is a step in the right direction, but consent alone isn't enough. True partnership requires sharing decision-making power, respecting cultural protocols, and ensuring that benefits flow to Indigenous communities. It requires acknowledging that de-extinction isn't just a scientific or ecological issue. It's a cultural and spiritual one. And it requires humility, the recognition that Western science doesn't have all the answers, and that Indigenous knowledge offers essential wisdom about our relationship with the natural world. The philosophical dimension of the debate touches on even deeper questions about nature, authenticity, and our place in the cosmos. Is a CRISPR-edited thylacine really a thylacine? Or is it a hybrid, a novel organism created by human intervention, bearing only superficial resemblance to its extinct ancestor? Philosopher Mark Segoff argues that de-extinction creates zombie species, genetic facsimiles that lack the evolutionary history, ecological context, and cultural meaning of the original. A revived thylacine won't have descended from the same lineage as historical thylacines. It won't have co-evolved with its ecosystem over millennia. It won't carry the same cultural significance for Indigenous peoples who knew the original species. It will be, in Segoff's words, a theme park version of nature, entertaining but inauthentic. This critique resonates with deeper anxieties about the Anthropocene, the geological epoch defined by human dominance over Earth's systems. In an age where we've altered the climate, reshaped continents, and rewritten genomes, what does "wild" even mean? Is there any nature left that hasn't been touched by human hands? De-extinction pushes this question to its logical extreme. If we can design species to our specifications, where do we draw the line between nature and artifact? Between conservation and creation? Between humility and hubris? These aren't just academic questions. They're existential ones that shape how we see ourselves and our place in the world. Do we see ourselves as gardeners tending a fragile planet? As engineers redesigning nature to suit our needs? As humble participants in a web of life we didn't create and can't fully control? Our answer determines not just whether we revive the thylacine, but how we approach every environmental challenge, from climate change to synthetic biology to space colonization. De-extinction forces us to confront these questions head-on, and there are no easy answers. But perhaps the most profound ethical question is this: does de-extinction represent hope or distraction? In an age of ecological grief, where every news cycle brings reports of species loss, habitat destruction, and climate disasters, it's tempting to see de-extinction as a beacon of hope. A reminder that we're not powerless. That we can fix what we've broken. That the future doesn't have to be defined by loss. This hope isn't trivial. Psychological research shows that hope is essential for motivating pro-environmental behavior. People who believe positive change is possible are more likely to take action than those who feel despair. De-extinction could inspire a new generation of conservationists, generate public support for broader environmental efforts, and demonstrate that humanity is capable of healing as well as harming. But hope can also be dangerous when it's misplaced. False hope, the belief that technological fixes will save us without behavioral change, can breed complacency. It can distract from the systemic changes needed to address root causes of the biodiversity crisis: overconsumption, inequitable resource distribution, short-term economic thinking. De-extinction risks becoming a conservation spectacle, a dramatic, but ultimately superficial intervention that makes us feel good without addressing deeper problems. The real work of conservation isn't glamorous. It's protecting habitat acre by acre. It's negotiating with farmers and loggers. It's changing policies and economic incentives. It's confronting uncomfortable truths about our lifestyles and values. De-extinction can't replace this work. At best, it can complement it if framed correctly. The ethical challenge is to harness de-extinction's inspirational power without letting it become a distraction. To use it as a gateway to deeper engagement with conservation, not an excuse to avoid hard choice. This requires honest messaging: de-extinction is a tool, not a solution. It addresses symptoms, not root causes. It can restore lost species, but it can't prevent future extinctions without broader systemic change. Framed this way, de-extinction becomes not a techno-fix, but a teaching tool, a visceral demonstration of both our power to destroy and our capacity to heal. It becomes a catalyst for deeper reflection about our relationship with nature. And that reflection might be its greatest value. Navigating these ethical complexities requires more than individual conscience. It requires robust governance frameworks that balance competing values, incorporate diverse perspectives, and adapt to new information. The Genetic Heritage Act's De-extinction Oversight Committee represents one model: multidisciplinary, transparent, accountable. But governance alone isn't enough. Ethical decision-making requires ongoing public dialogue, not just expert deliberation behind closed doors. It requires creating spaces where scientists, ethicists, indigenous knowledge holders, conservationists, farmers, and ordinary citizens can grapple with these questions together. It requires acknowledging that there are no perfect answers, only better and worse choices made with humility, transparency, and a commitment to learning from mistakes. It requires recognizing that ethics isn't a barrier to progress. It's the foundation of responsible innovation. Technologies without ethical guardrails become dangerous. De-extinction without ethical reflection risks repeating the very mistakes that caused extinctions in the first place: arrogance, short-sightedness, disregard for complex relationships. The goal isn't to stop de-extinction. The goal is to do it wisely, to ensure that our power to resurrect is matched by our wisdom to know when, how, and why to use it. As the first Thylacines prepare for release, these ethical questions remain unresolved, not because we lack answers, but because the questions themselves evolve as the science advances. Each success raises new dilemmas. Each failure teaches new lessons. The ethical landscape shifts with every decision. This isn't a weakness. It's a strength. It reflects the humility required to navigate uncharted territory. De-extinction isn't a destination. It's a journey, one that requires us to walk slowly, listen carefully, and adjust our course as we learn. The Thylacines' return isn't just an ecological experiment. It's an ethical one. It tests not just our scientific capabilities, but our moral imagination. Can we revive a species without repeating the arrogance that caused its extinction? Can we heal ecological wounds without creating new ones? Can we wield godlike power with human humility? These questions have no final answers. But asking them, and taking them seriously, is what separates responsible stewardship from reckless ambition. The Thylacine is waiting. The ethical crossroads is before us. Which path we choose will say more about us than about the species we seek to revive. And that choice, more than any technological breakthrough, will determine whether de-extinction becomes a force for healing or a cautionary tale for future generations. The image is seared into public consciousness: a lone Thylacine pacing behind bars, visitors pressing against glass, flash photography capturing its every movement. This is the fate many imagine for de-extinct species: a life of captivity, a museum exhibit, a living relic of human failure. But this image is wrong. The Genetic Heritage Act explicitly prohibits keeping revived species in captivity except for temporary quarantine or medical treatment. Section 1 UK mandates that all de-extinct animals must be released into protected wild habitats, areas where they can fulfill their ecological roles, express natural behaviors, and contribute to ecosystem function. This isn't just an ethical preference. It's a legal requirement. And it reflects a fundamental shift in conservation philosophy. From preservation to restoration, from captivity to rewilding, from spectacle to stewardship. But this raises a critical question: where, exactly, will these animals live? Tasmania in 2025 and Tasmania in 1936. The landscape has been transformed by logging, agriculture, urbanization, and invasive species. Suitable habitat has been fragmented, degraded, or lost entirely. Releasing a thylacine into the wild isn't as simple as opening a cage door. It requires restoring ecosystems, securing protected areas, mitigating threats, and building community support. It requires creating not just space for individual animals, but functional landscapes where entire ecological communities can thrive. This isn't just about finding somewhere to put revived species. It's about healing the places they'll call home. The first challenge is habitat availability. Historical records show that thylacines once reigned across mainland Australia and Tasmania, occupying diverse habitats from coastal forests to alpine regions. But by the early 20th century, their range had contracted to Tasmania's wilderness areas, particularly the western and southwestern regions where rugged terrain and low human density provide refuge. Today, these areas remain the most suitable for reintroduction, but they're not pristine. Logging operations have fragmented old-growth forests. Mining exploration has disturbed remote areas. Climate change has altered fire regimes and rainfall patterns. Invasive species like feral cats, red foxes, and European rabbits have established themselves even in the most remote wilderness. Releasing thylacines into these landscapes without addressing these threats would be irresponsible, like sending soldiers into battle without weapons. The reintroduction plan therefore includes a comprehensive habitat restoration component, removing invasive predators from release areas, restoring native vegetation through replanting and controlled burns, creating wildlife corridors to connect fragmented habitats, and establishing long-term monitoring programs to track ecosystem health. This restoration work began years before the first thylacine embryo was created. In 2020, the Tasmanian government designated Pigeon, hectares of western wilderness as the Thylacine recovery zone, an area where logging, mining, and development would be restricted to prioritize ecological restoration. Since then, conservation groups have removed over 1 feral cats from the zone, replanted 2 hectares of native vegetation, and established kilometers of wildlife corridors connecting isolated habitat patches. This isn't just preparation for thylacine release. It's ecosystem healing that benefits countless other species, devils, quolls, padmelons, and the plants and insects that depend on healthy forests. The thylacine is the flagship, but the beneficiaries are the entire ecological community. But habitat restoration alone isn't enough. Protected areas must be legally secured and effectively managed. Tasmania already has an extensive protected area network, national parks, nature reserves, and World Heritage areas covering over half of the island. But these protections aren't uniform. Some areas allow limited logging or mining. Others lack adequate funding for management and enforcement. Still others are too small or isolated to support viable populations of wide-ranging predators like thylacines. The reintroduction plan therefore calls for expanding and strengthening protected areas in the thylacine recovery zone. This includes upgrading existing reserves to strict protection status where all extractive activities are prohibited, creating new reserves in critical habitat areas, and establishing buffer zones around core protected areas where sustainable land uses are permitted but intensive development is restricted. These legal protections are essential for long-term success. A thylacine released today could live for 1 or more years in the wild. Its offspring could live for decades. The population could persist for centuries if the habitat remains protected. But legal protections alone aren't sufficient. Effective management requires adequate funding, trained staff, monitoring programs, and community engagement. Tasmania's Parks and Wildlife Service has historically been underfunded, with limited capacity to manage existing protected areas, let alone new ones. The reintroduction plan addresses this through a dedicated Thylacine Stewardship Fund, a $1 million endowment established through government contributions, philanthropic donations, and revenue from sustainable ecotourism. The fund will provide permanent financing for habitat management, threat control, monitoring, and community programs in the recovery zone. This financial sustainability is critical. Conservation projects often fail when initial enthusiasm fades and funding dries up. The endowment ensures that thylacine habitat will be protected and managed for generations to come, not just for the first few years after release. Wildlife corridors are another critical component of the reintroduction strategy. Thylacines are wide-ranging predators with historical home ranges of 2 square kilometers for males and 20 square kilometers for females. A viable population requires thousands of square kilometers of connected habitat, not isolated patches separated by roads, farms, and towns. But Tasmania's landscape has been fragmented by human development. Major highways like the Lille Highway cut through potential thylacine habitat, creating barriers to movement and increasing vehicle strike risk. Agricultural areas and plantations further fragment the landscape. To address this, the reintroduction plan includes an ambitious wildlife corridor program, constructing wildlife overpasses and underpasses on major highways, restoring riparian corridors along rivers and streams, creating stepping stone habitats in agricultural landscapes, and working with landowners to establish conservation covenants on private property. The goal is to create a connected network of habitats where thylacines and other wildlife can move freely across the landscape. This connectivity is essential not just for thylacines, but for ecosystem resilience in the face of climate change. As temperatures rise and rainfall patterns shift, species will need to move to track suitable conditions. Fragmented landscapes trap species in place, increasing extinction risk. Connected landscapes allow species to adapt and persist. The Thylacine Corridor program therefore serves multiple purposes: enabling thylacine movement, reducing road kill, supporting biodiversity, and building climate resilience. It's an investment in landscape-scale conservation that will benefit countless species for centuries to come. But perhaps the most innovative aspect of the reintroduction plan is its integration of indigenous land management. For Aboriginal Tasmanians, the land isn't just a resource to be managed. It's a living relative, a teacher, a source of identity and belonging. Their relationship with country is based on reciprocity, responsibility, and deep knowledge accumulated over tens of thousands of years. This knowledge offers invaluable insights for thylacine habitat restoration. Aboriginal cultural burning practices, low-intensity fires applied at specific times and places, can reduce fuel loads, promote diverse vegetation structure, and create mosaic landscapes that support both predators and prey. Traditional ecological knowledge identifies areas where thylacines were historically abundant, areas that might be suitable for reintroduction today. Indigenous land management principles emphasize working with natural processes rather than against them, building resilience rather than controlling outcomes. The reintroduction plan recognized this wisdom by establishing Aboriginal-led, caring for country programs in the Thylacine Recovery Zone. Aboriginal rangers will manage habitat restoration, conduct cultural burns, monitor wildlife populations, and share traditional knowledge with scientists and land managers. They'll also develop cultural protocols for engaging with the revived thylacine, protocols that honor the species' spiritual significance while ensuring its ecological role is fulfilled. This partnership isn't just ethical. It's practical. Indigenous knowledge complements Western science, filling gaps that data alone can't address. Together, they create a more holistic, culturally grounded approach to landscape restoration. The Thylacine's return isn't just about restoring a species. It's about restoring relationships with the land, with indigenous peoples, with our own place in the web of life. Community engagement is equally critical for long-term success. Releasing thylacines into the wild won't work if local communities oppose it. Farmers worry about livestock predation. Hunters worry about competition for game. Residents worry about safety. These concerns are legitimate and must be addressed through transparent communication, collaborative planning, and tangible benefits. The reintroduction plan includes a comprehensive community engagement strategy, public education campaigns explaining the ecological benefits of thylacines, particularly their role in controlling feral cats and foxes that also prey on livestock, compensation programs for verified livestock losses, citizen science programs engaging locals in monitoring and research, and economic opportunities through sustainable ecotourism. The goal isn't to convince everyone to support thylacine reintroduction. It's to build a broad coalition of supporters while addressing legitimate concerns of opponents. This requires listening more than lecturing, collaborating more than commenting, and demonstrating respect for diverse perspectives. In areas where opposition remains strong, the plan includes adaptive management, delaying releases, modifying approaches, or finding alternative solutions. Community support isn't a nice-to-have. It's essential. History is littered with conservation failures caused by top-down approaches that ignored local knowledge and concerns. The Thylacine program is determined not to repeat those mistakes. Success depends on building genuine partnerships with the people who live alongside thylacines, not just securing their permission, but earning their active support. Monitoring and adaptive management complete the reintroduction framework. Releasing thylacines into the wild isn't a one-time event, it's the beginning of a decades-long commitment to learning, adjusting, and stewarding. Every released individual will be fitted with EEPS collars and camera traps to track movements, survival, reproduction, and ecological impacts. Scientists will monitor changes in prey populations, predator activity, vegetation structure, and biodiversity across multiple trophic levels. This data will be publicly available through an online dashboard, allowing scientists, policymakers, and the public to track progress in real time. But monitoring alone isn't enough. The program must be prepared to adapt based on what it learns. If thylacines struggle to survive, additional support may be needed: supplementary feeding, veterinary care, or habitat modifications. If they cause unexpected harm, preying heavily on endangered species, for example, management strategies may need to be adjusted or releases paused. If community concerns arise, engagement efforts may need to be intensified. This adaptive approach reflects the humility required for responsible reintroduction. We don't have all the answers. We're learning as we go. And we're committed to adjusting our course based on evidence and feedback. This isn't weakness. It's strength. It acknowledges uncertainty while allowing progress. It builds trust through transparency and accountability. And it ensures that the thylacines return serves ecological goals without causing unintended harm. Lessons from other reintroduction programs inform this approach. The gray wolf reintroduction to Yellowstone National Park offers both inspiration and caution. Wolves reduced overabundant elk populations, allowing willow and aspen to regenerate, which created habitat for beavers, songbirds, and fish. River morphology changed as vegetation stabilized banks. Biodiversity increased across multiple trophic levels. But the program also faced challenges: livestock conflicts, illegal killings, political opposition. Success required decades of planning, stakeholder engagement, adaptive management, and conflict mitigation. Closer to home, the eastern quoll reintroduction to mainland Australia demonstrates the importance of pre-release training in genetic diversity. Quolls that practiced hunting live prey before release had significantly higher survival rates. Populations founded by individuals from multiple source populations were more resilient. These lessons are being applied directly to the thylacine program. But not all reintroductions succeed. The Arabian oryx reintroduction to Oman initially succeeded but collapsed due to poaching and habitat loss. The golden lion tamarin release in Brazil required decades of habitat restoration and corridor creation to achieve modest success. These failures aren't reasons to avoid reintroduction. They're reasons to do it better, to learn from past mistakes, plan more thoroughly, engage stakeholders more deeply, and monitor more rigorously. The thylacine program is benefiting from this collective wisdom. It's not reinventing the wheel. It's building on decades of hard-won experience. The ultimate vision extends far beyond thylacines. The Thylacine Recovery Zone is conceived as a model for landscape-scale conservation, a place where ecological restoration, indigenous stewardship, community engagement, and scientific monitoring come together to heal damaged ecosystems. The lessons learned here will inform conservation efforts across Tasmania and beyond. The wildlife corridors created for thylacines will benefit countless other species. The habitat restoration work will improve ecosystem health for generations. The partnerships with indigenous communities will strengthen cultural connections to country. The community engagement programs will build broader support for conservation. The monitoring data will advance scientific understanding of ecosystem dynamics. This isn't just about one species. It's about creating a template for healing landscapes worldwide. In an age of biodiversity loss and climate change, we need more than isolated protected areas. We need connected, resilient, actively managed landscapes where humans and nature can coexist. The thylacines return offers an opportunity to demonstrate what this looks like in practice, not as a theoretical ideal, but as a lived reality. It's a bold vision. But bold visions are what conservation needs right now. As the first thylacines prepare for release, the landscape is waiting, not in its historical state, but in a state of becoming. Healing. Transforming. The forests are regenerating. The corridors are connecting. The communities are engaging. The indigenous knowledge is being shared. This isn't a return to the past. It's a step toward a different future, one where humans take responsibility for healing the damage we've caused, where conservation is about restoration rather than preservation, where landscapes are managed not just for their ecological value but for their cultural and spiritual significance. The thylacine won't live in a zoo. It will live in a restored wilderness, a place shaped by human hands, but guided by ecological wisdom, by indigenous knowledge, by community commitment. It will live in a landscape that remembers its past but isn't bound by it, a landscape that honors loss but chooses hope. A landscape that acknowledges human impact but redirects that impact toward healing. This is the promise of rewilding. Not perfection. Not purity. But possibility. The possibility of healing what we've broken. The possibility of living in right relationship with other species. The possibility of creating landscapes where both humans and wildlife can thrive. The Thylacine is waiting. The landscape is ready. The question is whether we're ready to welcome it home, not as a captive spectacle, but as a world neighbor in a restored world. The most persistent criticism of de-extinction isn't about ethics or feasibility. It's about opportunity cost. In a world losing species at 1,000 times the natural rate, is it responsible to spend millions reviving one extinct animal while thousands of living species slide toward oblivion? This isn't a rhetorical question. It's a mathematical one. The odd $2 million de-extinction fund could protect habitat for dozens of critically endangered species. The scientific expertise devoted to Thylacine genome editing could advance conservation genetics for living marsupials. The public attention generated by resurrecting the dead could be redirected toward saving the living. This tension between revival and prevention, between charismatic extinctions and quiet disappearances, lays at the heart of the conservation paradox. And resolving it requires moving beyond simplistic either or thinking toward a more nuanced understanding of how de-extinction fits within broader conservation strategy. The truth is that de-extinction isn't inherently helpful or harmful to living species. Its impact depends entirely on how it's implemented, whether it complements existing efforts or competes with them, whether it generates new resources or diverts existing ones, whether it inspires broader engagement or creates distraction. The evidence so far suggests that de-extinction can be a net positive for conservation, but only if deliberately designed to serve that purpose. The concern about resource diversion is legitimate. Conservation has always been underfunded. The global funding gap for biodiversity protection is estimated at $2 billion annually. Every dollar spent on de-extinction is a dollar not spent on habitat protection, anti-poaching efforts, or invasive species control. But this framing assumes a zero-sum game, that de-extinction funding comes directly from traditional conservation budgets. In reality, the relationship is more complex. De-extinction has attracted significant new funding from sources that previously didn't support conservation: technology philanthropists, biotech companies, private investors interested in genetic engineering applications. The TIGRR Lab's initial funding came from Silicon Valley donors who had never before contributed to wildlife conservation. The Revive and Restore initiative attracted corporate sponsorships from companies like Colossal Biosciences that see commercial potential in genetic rescue technologies. This new money argument is compelling but incomplete. While de-extinction has generated additional funding, it hasn't eliminated competition for existing resources. Conservation organizations still face difficult choices about where to allocate limited staff time, political capital, and public attention. A media cycle dominated by Thylacine embryos is a media cycle not dominated by critically endangered frogs. A policymaker excited about de-extinction technology might deprioritize habitat protection legislation. These opportunity costs are real, even if the financial costs aren't zero-sum. The challenge is to maximize the synergies while minimizing the trade-offs, to ensure that de-extinction amplifies rather than diminishes support for traditional conservation. One way de-extinction can amplify conservation is through technological spillover. The genetic tools developed for Thylacine revival, CRISPR editing protocols, stem cell differentiation techniques, artificial womb technologies, are already being applied to save living endangered species. The black-footed ferret, once reduced to just 7 individuals, now benefits from genetic rescue techniques that increase diversity in captive breeding programs. The northern white rhino, with only 2 females remaining, is the focus of efforts to create embryos from stored genetic material, a direct application of de-extinction technology. The Tasmanian devil, devastated by facial tumor disease, is being protected through genomic screening that identifies disease-resistant individuals for breeding. These applications aren't hypothetical. They're happening now, funded in part by de-extinction research budgets, and developed by scientists who work across both fields. Dr. Marilyn Renfree of the University of Melbourne explains, the same stem cell techniques we're developing for thylacines are being used to preserve genetic diversity in critically endangered marsupials like the mountain pygmy possum. We're not choosing between de-extinction and conservation. We're using de-extinction to advance conservation. This technological synergy is perhaps the strongest argument for de-extinction's conservation value. It transforms what could be a distraction into a catalyst, generating tools, expertise, and funding that benefit living species while pursuing the longer-term goal of species revival. Public engagement offers another potential synergy. Conservation has struggled for decades to capture public imagination. Habitat loss, climate change, and biodiversity decline are complex, slow-moving crises that lack the emotional punch of charismatic megafauna. De-extinction changes that equation. The thylacine is a powerful symbol, a ghost haunting Australia's ecological conscience, a reminder of human-caused extinction that resonates across generations. Reviving the thylacine isn't just about restoring one species. It's about telling a story, one of loss, responsibility, and redemption, that can inspire broader conservation action. Early evidence supports this. Since the Genetic Heritage Act passed, donations to traditional conservation organizations in Australia have increased by 1/6. Volunteer applications for habitat restoration projects have doubled. School programs focused on biodiversity have seen enrollment surge. The thylacine has become a gateway species, not in the ecological sense, but in the educational one. People who learn about thylacine de-extinction often become curious about broader conservation issues. They ask, what other species are at risk? What can I do to help? How does habitat loss affect wildlife? This gateway effect is difficult to quantify, but impossible to ignore. Conservation biologist Jurassic Martin of the University of Queensland notes, "We've been trying to get people excited about saving the orange-bellied parrot for 20 years. The thylacine has done in 2 years what we couldn't do in 2 decades. Is that a distraction? Or is it an opportunity we'd be foolish to ignore?" The challenge is to harness this attention intentionally, to use thylacine stories as entry points to broader conservation education, to connect revival efforts to habitat protection campaigns, to demonstrate that preventing extinction is always preferable to reversing it. But the gateway effect has limits. Charismatic species have always dominated conservation attention, the panda problem, where cute mammals receive disproportionate funding compared to less glamorous, but equally important species like insects, fungi, or plants. De-extinction risks amplifying this bias. Why focus on reviving the thylacine when the western ground parrot has fewer than just individuals left? Why invest in passenger pigeon revival when the regent honeyeater is down to shoes birds? These aren't rhetorical questions. They reflect a genuine concern that de-extinction could reinforce existing inequities in conservation funding and attention. The response requires deliberate effort to balance charismatic revival projects with support for less visible, but equally critical conservation needs. The Genetic Heritage Act attempts this through its biodiversity multiplier requirement. De-extinction projects must demonstrate net positive impact on overall biodiversity, not just the revival of a single species. The Thylacine program, for example, has committed lots of its funding to habitat restoration that benefits dozens of co-occurring species. It has established monitoring programs that track ecosystem health across multiple taxa. It has partnered with organizations working on less charismatic but critically endangered species. These efforts don't eliminate the bias entirely, but they mitigate it, ensuring that de-extinction serves broader conservation goals rather than competing with them. The policy dimension adds another layer of complexity. De-extinction has already influenced conservation policy in Australia. The Genetic Heritage Act includes provisions requiring increased funding for traditional conservation efforts proportional to de-extinction spending, a no net loss guarantee that prevents revival projects from cannibalizing existing budgets. It mandates public education campaigns emphasizing that prevention is always preferable to revival. It establishes oversight mechanisms to ensure that de-extinction complements, rather than undermines, broader biodiversity goals. These policy safeguards are crucial, but they're not sufficient. Conservation policy operates at multiple levels: federal, state, local, international, and de-extinction's influence will vary across these scales. At the international level, Australia's leadership on de-extinction governance has inspired similar frameworks in other countries. The European Union is drafting its own de-extinction guidelines, explicitly setting the Australian model. The Convention on Biological Diversity has established a working group on genetic rescue technologies. These developments could strengthen global conservation governance, but only if de-extinction frameworks prioritize ecological integrity over technological novelty. At the local level, de-extinction has sparked conversations about land use, habitat protection, and indigenous stewardship that might not have happened otherwise. Farmers who once saw conservation as a threat are now engaging with habitat restoration because of thylacine reintroduction plans. Loggers are participating in sustainable forestry discussions because of wildlife corridor requirements. These policy shifts aren't automatic. They require deliberate effort to connect de-extinction to broader conservation agendas. But when successful, they demonstrate that revival efforts can catalyze systemic change beyond their immediate scope. Measuring the net impact of de-extinction on conservation remains challenging. Traditional metrics, funding dollars, species counts, habitat acres, capture only part of the picture. The deeper question is whether de-extinction changes the trajectory of conservation itself, whether it shifts mindsets from preservation to restoration, from despair to hope, from isolation to collaboration. Early indicators are promising. The Thylacine program has fostered unprecedented collaboration between geneticists, ecologists, indigenous knowledge holders, and community groups. It has generated public interest in conservation that transcends traditional demographic boundaries. It has demonstrated that ambitious, long-term conservation goals are achievable with sustained commitment. But these benefits must be weighed against genuine risks, the potential for moral hazard, the danger of technological distraction, the possibility that revival efforts could fail and undermine public trust in conservation. The conservation paradox isn't a problem to be solved once and for all. It's a tension to be managed continuously, to be navigated with humility, transparency, and a commitment to learning from both successes and failures. De-extinction isn't a silver bullet for the biodiversity crisis. But it can be a valuable tool in the conservation toolkit if used wisely, integrated thoughtfully, and evaluated rigorously. The question isn't whether de-extinction helps or hurts living species. The question is whether we have the wisdom to ensure it helps more than it hurts. And that question will be answered not in laboratories or legislative chambers, but in the landscapes where conservation happens every day. For Aboriginal Tasmanians, the Thylacine, Lula or Kara, was never just an animal. It was a totemic being, a cultural symbol, a thread in the fabric of their ancestral identity. Its disappearance in Mungtor's Ortzalta wasn't just an ecological loss. It was a spiritual one inflicted by colonization, a severing of relationships that had existed for tens of thousands of years. So when scientists announced plans to revive the Thylacine using CRISPR gene editing, Aboriginal communities didn't react with simple enthusiasm or opposition. They responded with complexity, nuance, and deep reflection, asking questions that Western science rarely considers. Is this the right time? Are we ready? Does the land want the Lula back? These questions reflect a worldview fundamentally different from Western conservation paradigms, one that sees humans as part of nature rather than separate from it, that emphasizes reciprocity rather than domination, that values spiritual relationships alongside ecological ones. Understanding indigenous perspectives on de-extinction requires moving beyond token consultation to genuine partnership, recognizing that Aboriginal peoples have unique relationships with extinct species that must inform how revival efforts proceed. The diversity of indigenous perspectives is striking. There is no single Aboriginal view on Thylacine revival. Opinions vary by community, by elder, by individual, shaped by cultural background, personal experience, and spiritual beliefs. Some elders welcome the opportunity to restore a severed cultural connection. Auntie Patsy Cameron, a Pakana woman and cultural heritage advisor, explains, "The Lula is part of our dreaming. Its stories are woven into our songs, our dances, our ceremonies. When it disappeared, a part of our culture disappeared with it. Bringing it back isn't just about ecology. It's about healing, a chance to restore something that was taken from us." For Auntie Patsy and others like her, the Thylacine revival represents cultural reclamation, a way to reconnect with ancestral knowledge, to pass stories to younger generations, to demonstrate that indigenous relationships with country can endure even profound loss. This perspective views de-extinction not as scientific intervention, but as cultural restoration, a continuation of indigenous stewardship that was interrupted by colonization. But other elders express deep reservations. Uncle Graham Paul, an Aboriginal elder from Tasmania, asks, "Who are we to decide whether the Lula should return? Have we done the ceremony? Have we asked the land if it's ready? Have we prepared ourselves spiritually for this responsibility?" His questions reflect a worldview where relationships with other beings are governed by protocol, reciprocity, and respect, not just technical feasibility. In this view, reviving a species without proper cultural preparation could be spiritually dangerous, like opening a door without knowing what lies on the other side. These reservations aren't opposition to science. They're demands for respect, for recognition that indigenous peoples have unique relationships with extinct species that must be honored through proper protocols, consultation, and consent. The concept of cultural safety is central to indigenous perspectives on de-extinction. For many Aboriginal Australians, the Thylacine's extinction was not a natural event but an act of colonial violence. European settlers systematically eradicated the species through bounties, hunting, and habitat destruction actions justified by a worldview that saw predators as vermin and indigenous knowledge as primitive. Reviving the Thylacine without addressing this history could perpetuate colonial patterns of appropriation and control. Who owns the Thylacine's DNA? Who has the right to play with ancestral spirits? What obligations come with reviving a being that was deliberately killed by colonizers? These aren't abstract questions. They're demands for justice, for recognition that de-extinction must be part of broader processes of truth-telling, reconciliation, and decolonization. Draima Lee, a Trolwulwe woman and environmental researcher, explains, "We can't talk about bringing back the Lula without talking about why it disappeared. We can't discuss genetic editing without acknowledging that our ancestors' knowledge was erased alongside the species itself. De-extinction has to be about more than science. It has to be about healing the wounds of colonization. This healing requires more than consultation. It requires sharing decision-making power, respecting cultural protocols, ensuring that benefits flow to indigenous communities, and acknowledging that Western science doesn't have all the answers. True partnership means recognizing that indigenous knowledge offers essential wisdom about our relationship with the natural world, wisdom that can guide de-extinction efforts toward greater cultural and ecological integrity." Cultural protocols for engaging with revived species are still being developed, but they're essential for ensuring that Thylacine revival honors indigenous relationships rather than appropriating them. These protocols might include welcoming ceremonies when the first Thylacines are released, smoking ceremonies to cleanse and protect the animals, sung lines and stories shared with younger generations to reconnect them with Lula knowledge, restrictions on who can interact with the animals and how, guidelines for naming revived individuals, processes for handling deaths and remains with cultural respect. These aren't add-ons to the scientific process. They're integral to it, ensuring that revival efforts are culturally grounded, spiritually appropriate, and relationally responsible. The Genetic Heritage Act's requirement for free, prior, and informed consent is a step in the right direction, but consent alone isn't enough. True partnership requires ongoing dialogue, shared governance, and mutual respect. Aboriginal rangers are already being trained as Thylacine monitors. Indigenous knowledge holders are serving on the De-extinction Oversight Committee. Cultural protocols are being developed collaboratively with elders and community leaders. This partnership isn't just ethical. It's practical. Indigenous knowledge of historical Thylacine behavior, habitat preferences, and seasonal movements complements Western science, filling gaps that data alone can't address. Together, they create a more holistic, culturally grounded approach to ecological restoration. The intergenerational dimension of indigenous perspectives adds another layer of complexity. For elders who grew up with stories of the Lula, revival represents a chance to heal intergenerational trauma, to restore something that was lost during their lifetimes. For younger Aboriginal Australians, revival offers an opportunity to reconnect with cultural knowledge that colonization disrupted, to learn songs, stories, and ceremonies that were nearly lost. But this intergenerational healing requires careful navigation. Some young people worry that focusing on extinct species distracts from urgent contemporary issues, housing, health, education, justice. They ask, why spend millions reviving the Lula when our communities lack basic resources? These concerns are valid and must be addressed through tangible benefits, training programs, employment opportunities, cultural revitalization projects that flow from de-extinction efforts. The Thylacine program has committed to creating Aboriginal-led conservation jobs, supporting cultural heritage initiatives, and ensuring that Indigenous communities benefit economically and culturally from revival efforts. But these commitments must be honored consistently, not just at the outset, but throughout the decades-long process of reintroduction and stewardship. Intergenerational justice requires that de-extinction serves not just ecological goals, but social and cultural ones as well. Land rights and sovereignty are inextricably linked to Indigenous perspectives on de-extinction. For Aboriginal Australians, country isn't just land. It's identity, law, culture, spirituality. The Thylacine's historical range overlaps with areas where native title claims are unresolved, where mining and logging operations continue despite Indigenous opposition, where cultural heritage sites are threatened by development. Reviving the Thylacine without addressing these underlying issues of land justice would be incomplete at best, harmful at worst. As Uncle Michael Mansell, a Palawa leader and legal scholar, states, you can't bring back the Lula on stolen land. You can't restore ecological relationships while denying Indigenous sovereignty. De-extinction has to be part of a broader struggle for land rights, for self-determination, for justice. This perspective challenges conservation to think beyond species and habitats to the political and historical contexts in which restoration occurs. It demands that de-extinction efforts support, not undermine, indigenous land rights, native title claims, and cultural heritage protection. The Thylacine program has begun addressing this through partnerships with Aboriginal land councils, support for native title applications in the Thylacine recovery zone, and advocacy for stronger cultural heritage protections. But these efforts must be sustained and expanded, recognizing that ecological restoration and indigenous justice are not separate goals, but interconnected parts of a larger vision for healing country and community. The spiritual dimension of indigenous perspectives often eludes Western understanding, but it's essential for respectful engagement. For many Aboriginal Australians, the Thylacine isn't just a biological entity. It's a spiritual being with agency, intention, and relationship to other beings, including humans. Reviving the Thylacine isn't just a technical challenge. It's a spiritual undertaking that requires proper ceremony, respect, and reciprocity. Some elders worry that CRISPR editing, manipulating DNA in a laboratory, violates the sanctity of life, treating ancestral beings as objects to be engineered rather than relatives to be honored. Others see genetic technology as a tool that, like fire or stone tools, can be used wisely or unwisely depending on intention and protocol. These spiritual concerns aren't anti-science. They're pro-relationship, emphasizing that how we engage with other beings matters as much as what we do. They remind us that de-extinction isn't just about restoring ecological functions. It's about restoring right relationships with the land, with other species, with our own humanity. This relational perspective offers a profound critique of Western conservation's tendency to objectify nature, to see species as resources to be managed rather than beings to be related to. Indigenous knowledge invites us to approach de-extinction not as engineers redesigning nature, but as relatives welcoming back a lost family member, with humility, respect, and love. Navigating these diverse perspectives requires humility, patience, and genuine partnership. There are no easy answers. There is no single indigenous position to be represented. There is only ongoing dialogue, listening more than speaking, learning more than teaching, respecting more than appropriating. The Thylacine program has made significant progress in this direction, but the work is far from complete. True partnership requires sharing power, not just seeking permission. It requires honoring indigenous knowledge as equal to Western science, not subordinate to it. It requires recognizing that de-extinction isn't just a scientific or ecological issue. It's a cultural and spiritual one, and indigenous peoples have essential wisdom to offer about how to proceed with wisdom and respect. As Auntie Patsy Cameron reflects, the Lula was our teacher. It taught us about balance, about respect, about our place in the web of life. If it returns, it will have lessons for us again, not just about ecology, but about how to live in right relationship with all beings. Are we ready to listen? This question, simple yet profound, captures the essence of indigenous perspectives on de-extinction. It's not about whether we can revive the Thylacine. It's about whether we're ready to receive it back, with humility, respect, and the wisdom to honor the relationships it represents. The answer will determine not just the success of revival efforts, but the quality of the world we create together. While the world watches Thylacine embryos develop in laboratories, a quieter revolution is already underway in conservation field stations, breeding facilities, and protected areas across the globe. The genetic technologies developed for de-extinction, CRISPR gene editing, stem cell differentiation, genomic screening, artificial reproduction, are already being applied to save living endangered species from extinction. This isn't science fiction. It's happening now. The black-footed ferret, once reduced to just 7 individuals, is benefiting from genetic rescue techniques that increase diversity in captive breeding programs. The northern white rhino, with only 2 females remaining, is the focus of efforts to create embryos from stored genetic material. The Tasmanian devil, devastated by facial tumor disease, is being protected through genomic screening that identifies disease-resistant individuals for breeding. These applications represent the most immediate and tangible benefit of de-extinction research, not the revival of extinct species, but the prevention of future extinctions. The tools developed for resurrection are already serving salvation, and this practical impact is transforming conservation from a reactive discipline focused on preserving remnants to a proactive one focused on restoring resilience. The black-footed ferret offers one of the clearest examples of genetic rescue in action. By Alta. This North American predator had been reduced to a single population of 18 individuals in Wyoming, all descendants of just 7 founders. This extreme genetic bottleneck left the species vulnerable to disease, reproductive failure, and developmental abnormalities. Traditional captive breeding could maintain the population, but it couldn't restore the genetic diversity needed for long-term survival. Then came genetic rescue. Scientists at Revive and Restore partnered with the ewes, fish and wildlife surveys to sequence the genomes of historical furret specimens preserved in museums, specimens that represented genetic lineages lost in the modern population. Using CRISPR technology developed for de-extinction research, they edited the genomes of living furrets to reintroduce these lost genetic variants. The result was a significant increase in genetic diversity equivalent to adding 4 new founders to the breeding program. This isn't just theoretical. Genetically rescued furrets have shown improved immune function, higher reproductive success, and greater resilience to disease. In ecological, the first CRISPR-edited furrets were released into the wild in Colorado, where they've established a thriving population. TIGRR Lab, Director of Conservation Genetics at San Diego Zoo Wildlife Alliance, explains, "We're not just preserving the black-footed furret. We're improving it, giving it the genetic tools it needs to survive in a changing world. This is conservation ecological." The furret program demonstrates that genetic rescue isn't about creating perfect replicas of historical populations. It's about equipping species with the genetic diversity they need to adapt and persist. And the technologies developed for de-extinction made this possible. The northern white rhino presents an even more urgent application of de-extinction technology. With only 2 females remaining, Pigeon and Pigeon, both unable to reproduce naturally, the species is functionally extinct in the wild. But extinction isn't inevitable. An international consortium led by the Leibniz Institute for Zoo and Wildlife Research has been working since ecological to create northern white rhino embryos using stored genetic material from deceased males. The process involves harvesting eggs from the remaining females, fertilizing them with frozen sperm from deceased males, and implanting the resulting embryos into southern white rhino surrogates. This isn't cloning. Its advanced assisted reproduction technique was developed through de-extinction research on other species. In ecological, the team successfully created 2 viable northern white rhino embryos, which are now stored in liquid nitrogen awaiting surrogate mothers. While challenges remain finding suitable surrogates, ensuring successful pregnancies, raising calves without maternal guidance, the progress is remarkable. Thomas Hildebrand, the project's lead reproductive biologist, notes, 10 years ago, this would have been impossible. The technologies we developed for de-extinction research, egg harvesting, in vitro fertilization, embryo cryopreservation made this feasible. We're not just saving the northern white rhino. We're creating a template for saving other species on the brink. This template is already being applied. The Sumatran rhino, with fewer than 80 individuals remaining, is now the focus of similar assisted reproduction efforts. The Javan rhino, down to 72 animals, is benefiting from genomic screening to identify the most genetically valuable individuals for breeding. These applications demonstrate that de-extinction technology isn't a distraction from conservation. It's a lifeline for species with no other hope. Disease resistance offers another critical application of genetic technologies. The Tasmanian devil has been devastated by devil facial tumor disease, DFT, a rare contagious cancer that has reduced populations by over 16 since its emergence in Alta. Traditional conservation approaches, captive breeding, with population monitoring, disease-free insurance populations slowed the decline but couldn't stop it. Then came genomic screening. Scientists at the University of Tasmania sequenced the genomes of thousands of devils, identifying genetic variants associated with disease resistance. They discovered that some individuals carried natural immunity variants that allowed them to survive DFT exposure. Using this knowledge, they established a selective breeding program focused on these resistant individuals. The result has been dramatic. Captive bred devils with resistant genotypes have been released into the wild, where they're surviving and reproducing despite ongoing disease presence. Wood populations in areas with high frequencies of resistant genotypes are showing signs of recovery. Catherine Belov, a geneticist leading the research, explains, "We're not editing devil genomes yet, that's still controversial. But we're using genomic information to guide breeding decisions, which is a form of genetic management." The technologies developed for de-extinction, high-throughput sequencing, bioinformatics analysis, genetic screening, made this possible. This approach is now being applied to other disease-threatened species, amphibians facing chytrid fungus, bats confronting white nose syndrome, Hawaiian honeycreepers battling avian malaria. Genetic technologies aren't just about editing DNA. They're about understanding it, and using that understanding to make smarter conservation decisions. Cryopreservation and biobanking represent another practical application of de-extinction research. The San Diego Zoo's Frozen Zoo, established in 1970, now holds genetic material from over 100 species and 100 individual animals, sperm, eggs, embryos, tissue samples preserved in liquid nitrogenate. This living library serves as an insurance policy against extinction, preserving genetic diversity that can be used for future breeding, research, or even revival efforts. But cryopreservation technology has advanced dramatically thanks to de-extinction research. Scientists have developed new techniques for freezing and thawing delicate cells like eggs and embryos without damage. They've created protocols for preserving stem cells that can be differentiated into any cell type. They've established international networks for sharing genetic material across borders and institutions. These advances are already paying dividends. In 2020, scientists used frozen sperm from a deceased male black-footed for it to inseminate a female, producing offspring with genetic diversity not seen in decades. In 2021, researchers thawed and cultured stem cells from the critically endangered Mississippi gopher frog, creating a renewable source of genetic material for future breeding. Barbara Durant, director of reproductive sciences at San Diego Zoo Wildlife Alliance, notes, "The frozen zoo isn't just a museum of genetic material. It's an active conservation tool. Every sample we preserve is a potential lifeline for a species on the brink. And the technologies we're developing through de-extinction research are making that lifeline stronger." Biobanking is now expanding globally, with new facilities opening in Australia, Brazil, and South Africa, ensuring that genetic diversity is preserved not just for charismatic megafauna, but for amphibians, insects, plants, and other overlooked but ecologically critical species. Assisted reproduction technologies developed for de-extinction are revolutionizing captive breeding programs worldwide. Artificial insemination, in vitro fertilization, embryo transfer, these techniques were once experimental. Now they're standard practice in zoos and breeding centers. The giant panda, once notoriously difficult to breed in captivity, now has a thriving captive population thanks to advanced reproductive technologies. The California condor, reduced to just 27 individuals in 2016, has been brought back from the brink through carefully managed breeding programs that use artificial insemination to maximize genetic diversity. The Przewalski's horse, extinct in the wild until reintroduction efforts began in the 1920s, now benefits from embryo transfer techniques that allow valuable genetics to be shared across continents without moving animals. These successes aren't accidental. They're the result of decades of research into reproductive biology, research accelerated by de-extinction efforts. Dr. Kumitzoli, a reproductive scientist at the Smithsonian Conservation Biology Institute, explains, "Every species has unique reproductive challenges. What works for pandas doesn't work for rhinos. What works for birds doesn't work for marsupials." De-extinction research forced us to develop species-specific protocols to understand the nuances of reproduction across the tree of life. Those protocols are now saving species we never intended to save. This cross-pollination of knowledge is perhaps the most valuable practical application of de-extinction technology, not the revival of extinct species, but the prevention of future extinctions through advanced reproductive management. Genomic monitoring is transforming how we track and manage wood populations. Traditional wildlife monitoring, camera traps, radio collars, visual surveys provides valuable data but has limitations. It can't detect genetic diversity, disease susceptibility, or adaptive potential. Genomic monitoring changes that. By analyzing NAF from non-invasive samples like feces, hair, or saliva, scientists can track population size, genetic diversity, relateness, and even individual health without disturbing animals. This technology is already being used to monitor critically endangered species, mountain gorillas in Rwanda, snow leopards in the Himalayas, vaquitas in the Gulf of California. In Tasmania, genomic monitoring of devil populations has revealed unexpected pockets of genetic diversity, populations that survive death through natural resistance. These discoveries have redirected conservation efforts, focusing protection on areas with high conservation value. Andrew Storfer, a conservation geneticist at Washington State University, notes, "Genomic monitoring isn't just about counting animals. It's about understanding their evolutionary potential, their capacity to adapt to future challenges like climate change or emerging diseases. This is the future of conservation, not just preserving what exists, but ensuring it can persist." The technologies enabling this monitoring, high-throughput sequencing, bioinformatics pipelines, field-deployable DNA analyzers were developed and refined through de-extinction research. What began as tools for resurrecting the dead are now essential for protecting the living. But these practical applications come with ethical considerations that can't be ignored. Genetic rescue, assisted reproduction, genomic monitoring, these technologies are powerful, but they're not neutral. They raise questions about intervention versus non-intervention, naturalness versus artificiality, short-term gains versus long-term consequences. Is it right to edit the genome of a world species, even to save it from extinction? Does assisted reproduction create animals that can't survive without human help? Does genomic monitoring reduce wildlife to data points, objectifying beings that should be respected as whole entities? These questions don't have easy answers. They require ongoing ethical reflection, transparent decision-making, and inclusive dialogue with diverse stakeholders, including indigenous communities, animal welfare advocates, and the public. The conservation community is grappling with these questions through initiatives like the IUCN's Guidelines for Conservation Translocations and the Genetic Control of Invasive Rodents Program's ethical framework. These efforts recognize that technology alone isn't enough. Responsible application requires ethical wisdom, cultural sensitivity, and ecological humility. De-extinction technology isn't a silver bullet. It's a tool, and like any tool, its value depends on how it's used. The most profound impact of de-extinction technology may be cultural, rather than technical. For decades, conservation has been framed as a story of loss, a narrative of species disappearing, habitats shrinking, hope fading. This narrative isn't wrong, but it's incomplete. It emphasizes what we're losing without highlighting what we can save. De-extinction technology offers a different narrative, one of agency, innovation, and hope. It demonstrates that humans aren't just destroyers. We're also healers. We have the capacity not just to mitigate damage, but to actively restore what's been lost. The shift in narrative is already influencing conservation practice. Young people who once saw conservation as a losing battle are now pursuing careers in genetic rescue, reproductive biology, and ecosystem restoration. Donors who were hesitant to fund doom and gloom conservation are now investing in proactive restoration efforts. Policymakers who viewed conservation as a cost are now seeing it as an investment in technological innovation and ecological resilience. Joseph Bennett, a conservation biologist at Carleton University, observes, de-extinction has changed the conversation. It's moved us from asking, how do we slow the decline, to how do we actively restore? That's a fundamental shift in mindset, and it's generating energy, resources, and innovation that benefit all of conservation. This cultural shift may be the most valuable practical application of de-extinction technology, not the tools themselves, but the hope they inspire, the belief that we can heal what we've broken, that extinction isn't always forever, that the future doesn't have to be defined by loss. As de-extinction technology continues to advance, its practical applications will expand. Gene drives to control invasive species. Synthetic biology to create disease-resistant crops that reduce pressure on world habitats. Microbiome engineering to restore soil health in degraded landscapes. These applications aren't science fiction. They're emerging now, built on the foundation of research initially developed for species revival. The line between de-extinction and conservation is blurring, not because de-extinction is replacing traditional approaches, but because it's enriching them. The tools developed for resurrection are becoming standard equipment in the conservation toolkit. And the most important lesson they're teaching us is this: conservation isn't just about preserving the past. It's about actively shaping the future. It's about using our knowledge, or technology, or compassion to create a world where both humans and wildlife can thrive. The Thylacine embryos in laboratories capture headlines, but the real revolution is happening quietly in field stations and breeding centers worldwide, where genetic technologies are already saving species from extinction, restoring ecosystems, and offering hope for a more resilient future. De-extinction isn't a distraction from conservation. It's a catalyst for its evolution. And that evolution is already underway. The word de-extinction was always a misnomer. It suggested a return to the past, a reversal of time, a restoration of what was lost. But that was never the goal. The Thylacine being developed in laboratories isn't a perfect replica of the Montour's animal. It's a hybrid, part historical genome, part edited sequences, part adapted to modern conditions. It won't descend from the same lineage. It won't carry the same cultural knowledge. It won't inhabit the same world. Calling this de-extinction created unrealistic expectations, fooled ethical concerns, and distracted from the real purpose, not resurrection, but restoration. Not bringing back the dead, but healing the living. As the field matures, a new language is emerging, one that better reflects the nuanced reality of what's happening. Genetic rescue. Ecological replacement. Functional restoration. These terms acknowledge that we're not recreating the past. We're creating something new, something informed by history, but shaped by present needs and future possibilities. This linguistic shift isn't just semantics. It's a fundamental reframing of the entire enterprise, from a nostalgic attempt to undo loss to a forward-looking effort to build resilience. And this reframing is essential for the future of conservation in the Anthropocene. The Anthropocene, the geological epoch defined by human dominance over Earth's systems, presents conservation with unprecedented challenges. Climate change is altering habitats faster than species can adapt. Habitat fragmentation is isolating populations, reducing genetic diversity. Invasive species are disrupting ecosystems worldwide. The current extinction rate is estimated at just about 2 times the natural background rate. Traditional conservation approaches, protected areas, captive breeding, habitat restoration, are necessary but insufficient. They're fighting a rearguard action against overwhelming forces. What's needed is a new paradigm, one that embraces active intervention, technological innovation, and ecological engineering. Genetic rescue offers a crucial component of this new paradigm. It doesn't replace traditional approaches. It enhances them, providing tools to increase genetic diversity, restore ecological functions, and build resilience in the face of rapid change. The Thylacine isn't being revived to recreate mammoths to Tasmania. It's being developed to restore predator-prey dynamics in a modern ecosystem facing mesopredator release and trophic downgrading. The passenger pigeon isn't being brought back to relive the century skis. It's being engineered to restore seed dispersal and forest disturbance regimes in eastern North American ecosystems altered by human activity. These aren't exercise in nostalgia. They're practical interventions designed to address contemporary ecological challenges. And they represent a fundamental shift in conservation philosophy, from preservation to restoration, from protection to active stewardship, from reactive defense to proactive healing. The shift requires rethinking our relationship with nature. For centuries, Western conservation has been shaped by the wilderness ideal, the belief that nature is purest when untouched by human hands, that our role is to protect pristine landscapes from human influence. This ideal has inspired magnificent achievements, national parks, wilderness areas, global conservation movements. But it's increasingly inadequate for the Anthropocene. There is no untouched nature left. Every ecosystem on Earth has been shaped by human activity, directly through land use, indirectly through climate change, historically through millennia of coevolution. The wilderness ideal creates a false dichotomy between humans and nature, positioning us as either destroyers or protectors, but never as participants. Genetic rescue challenges this dichotomy. It acknowledges that humans are already shaping evolution through habitat alteration, climate change, and species introductions. The question isn't whether we should intervene. We already are. The question is whether we can intervene wisely, with humility, knowledge, and respect for ecological complexity. This isn't about playing God. It's about accepting responsibility for the power we already wield. It's about using our knowledge and technology not to dominate nature, but to heal it. Indigenous worldviews offer valuable guidance here. For many indigenous cultures, humans aren't separate from nature. We're participants in a web of relationships, with responsibilities to other beings, to the land, to future generations. Genetic rescue, when approached with this relational mindset, becomes not an act of domination, but an expression of stewardship, a way of using our gifts to care for the world we share with other species. The future of genetic rescue lies not in spectacular revivals of charismatic extinct species, but in quiet, practical applications that support living biodiversity. The technologies developed for Thylacine revival are already being used to save the mountain pygmy possum from climate change, to protect the orange-bellied parrot from disease, to restore genetic diversity in fragmented koa populations. These applications won't make headlines. They won't capture public imagination like resurrecting the dead. But they're where the real conservation impact lies, in the daily work of protecting species, restoring ecosystems, building resilience. The future of genetic rescue is integration, becoming a standard tool in the conservation toolkit, used alongside habitat protection, invasive species control, and climate adaptation. It's normalization, moving from experimental novelty to routine practice, from specialized laboratories to field stations and breeding centers worldwide. It's democratization, making genetic technologies accessible to conservationists in developing countries, where biodiversity is richest but resources are scarcest. Organizations like the African Conservation Genetics Network are already working toward this goal, training local scientists, establishing regional biobanks, developing species-specific protocols for African wildlife. This democratization is essential for ensuring that genetic rescue serves global conservation needs, not just the priorities of wealthy nations. Ethical frameworks will need to evolve alongside the technology. Current guidelines, like the IUCN's guidelines for reintroductions and the Genetic Heritage Act's oversight provisions, are valuable starting points, but they're based on a precautionary principle that may not be sufficient for the Anthropocene's urgent challenges. We need ethical frameworks that balance precaution with pragmatism, that recognize the moral imperative to act in the face of extinction crisis, that prioritize ecological function over genetic purity, that center indigenous knowledge and community consent. These frameworks must be living documents, continuously updated as science advances, as societal values evolve, as we learn from both successes and failures. They must be inclusive, incorporating diverse perspectives from scientists, ethicists, indigenous knowledge holders, animal welfare advocates, and the public. And they must be humble, acknowledging that we don't have all the answers, that uncertainty is inherent in ecological intervention, that our responsibility is to learn, adapt, and improve over time. The future of genetic rescue depends not just on technological advancement, but on ethical maturity, on our ability to wield powerful tools with wisdom, compassion, and respect. Education and public engagement will be critical for building support and ensuring responsible application. Genetic rescue is complex, nuanced, and often misunderstood. Media coverage tends to focus on sensational aspects, Jurassic Park scenarios, playing God narratives, while ignoring the practical, conservation-focused applications. This misrepresentation fuels public skepticism and policy resistance. Changing this narrative requires deliberate effort, clear communication about what genetic rescue is and isn't, transparent sharing of both successes and failures, inclusive dialogue that invites public participation in decision-making. Schools can integrate genetic rescue into biology curricula not as science fiction, but as real-world conservation science. Museums can create exhibits that explain the technology, its applications, and its ethical dimensions. Citizen science programs can engage the public in monitoring revived populations and restored ecosystems. These efforts won't eliminate controversy. Genetic rescue will always raise difficult questions, but they can build a more informed, nuanced public discourse. And that discourse is essential for ensuring that genetic rescue serves the public good rather than private interests or technological enthusiasm. The ultimate goal of genetic rescue isn't to create a menagerie of revived species. It's to restore ecological function, to rebuild resilience, to heal damaged ecosystems. The Thylacine's value isn't in its genetic authenticity. It's in its role as Tasmania's apex predator, regulating herbivore populations, suppressing mesopredators, maintaining ecosystem balance. The passenger pigeon's value isn't in its historical abundance. It's in its function as a seed disperser and forest disturber, shaping eastern North American ecosystems in ways that benefit countless other species. Genetic rescue succeeds not when we create perfect replicas of extinct animals, but when we restore the ecological relationships they represented. This functional approach shifts the focus from species to systems, from authenticity to effectiveness, from nostalgia to resilience. It acknowledges that ecosystems are dynamic, not static, that they've always changed, and will continue to change. Our role isn't to freeze them in time, but to guide their evolution toward greater health, diversity, and resilience. Genetic rescue is a tool for that guidance, not a return to the past, but a step toward a more vibrant, functional future. As we stand at the threshold of this new era in conservation, we face a choice. We can cling to the wilderness ideal, watching helplessly as ecosystems degrade and species disappear. Or we can embrace our role as active stewards, using our knowledge, or technology, or compassion to heal the damage we've caused and build a more resilient world. Genetic rescue isn't a panacea. It won't solve the biodiversity crisis alone. It must be part of a broader strategy that addresses root causes, habitat destruction, climate change, overconsumption, inequity. But it can be a powerful component of that strategy, a way to restore what's been lost, to strengthen what remains, to build hope in the face of despair. The Thylacine embryos in laboratories aren't about reversing history. They're about shaping the future. They're a testament to human ingenuity, yes, but also to human responsibility. They remind us that we have the power to destroy, but also the capacity to heal. They challenge us to use that power wisely, to act not out of arrogance, but out of love for the world we share with other species. The future of conservation isn't about choosing between preservation and intervention, between wilderness and technology, between past and future. It's about integrating all these elements into a new paradigm, one that honors the past, engages the present, and builds hope for the future. Genetic rescue is part of that paradigm. Not as a spectacle, but as a service. Not as resurrection, but as restoration. Not as an end, but as a means, a tool for healing the world, one species, one ecosystem, one relationship at a time. The Thylacine is waiting. The future is calling. The choice is ours.